Thank you so much. As Dr. Sanjay has a flight to catch, so I would request the chairperson, Dr. Pawan Jacob, sir, to please felicitate him with a memento. Thank you so much, sir. Now, I feel privileged to invite our next knowledgeable speaker of the day, Professor Farhan Jalil Ahmed, sir. Sir is currently a professor in School of Pharmaceutical Education and Research, Jamia Hamdard. He is an internationally known researcher in the area of pharmaceutical sciences with experience of industrial research through his fruitful association with Ranbaxy Research Laboratories for 6 long years as a scientist. The focus of his group has been in the area of nanomedicine, and many of his nano products have been approved by the DCGI and successfully transferred to the defense services for the use in the armed forces. He has published more than 450 publications and has 29 patents to his credit. He has earned many awards, including Young Scientist Fast Track from DST, Fast Track Scientist of the Year 2005 from NESA, UGC Research Award, Bharat Jyoti Award, Pharma Ratna Award 2017, ABEP Senior Scientist Award 2017. Now I would request sir to please enlighten us with his wise words.
A very good strength, and we have a wonderful first in time. So in this absence, I am presenting the presentation. Right. So I will start with a very famous quote from Mr. Dhirubhai Ambani, renowned industrialist. If you don't build your dream, someone else will hire you to help them build their own dreams. So as a young researcher, your mind, your brain is very fertile, very open. As we grow older, as we become senior, we become more focused. We are focused on one particular area. Now in my age, I only know the subject in which I am working. But your age, you are exposed to variety of subjects. You are exposed to variety of ideas. You are interacting, you are socializing. You have so many platforms where you can socialize, you can share your information, you can share your ideas. So whatever idea you have, never be ashamed of any thought, any idea, anything which is coming into your mind related to your work, related to your own future plan. All those things like Mr. Dhirubhai Ambani he did, he started from scratch, and you know today what he has created. So you all can dream, and you should be positive about it. That whatever dream I am thinking about, it can be converted into a reality. You don't have to be a big industrialist. You don't need very high amount of funds to convert your product into your thought or your dream into a product or technology or some process or some protocol which can benefit the society and which can also give you some financial returns. Another quote. So, what is the entrepreneur? If you see, the incubation process allows students, entrepreneurs, or student startups to preserve their capital gain by giving various types of supports. These supports are basically, if you see, what is a support which you are looking for. Very good example when last time I presented given by Dr. Saurav, that incubation center is like bungee jumping. If I ask you to climb a cliff and jump from there or from a high-rise building, if I ask you to jump, what will happen? That is suicide. You will die. But everybody loves bungee jumping. There also you are jumping from the cliff from a high, but because you have that rope which is attached to your feet, you know that you will not die. You enjoy it. You have something to support. Similarly, a university-based incubation center is like bungee jumping, where without very heavy investment, when you are investing you're not very heavily investing, you're only investing your intellect, investing your time, and you are getting different types of support. What is the support? Support like technical guidance, support like financial guidance, support like how to register your technology, how to take it forward, how to transfer from lab to scale up to field level to marketing. All those supports will be provided to students interfere in that incubation center. We have mentors, which we have included in the center. Mentors are basically successful industrialists, those who are our friends. Mentors are our own alumni who have successfully developed their own companies, their own startups. Mentors are some other banks and financial advisors who can tell you how to minimize your losses, how to plan your financial map. Because maybe you are a technologist, maybe you are a biotechnology student, maybe you are a biochemistry student and you are not aware how to plan map, how to plan my finances, how to plan my strategy. So those things, those strategies, those help will be provided free of charge in the incubation center. So if you go by definition, business incubation is a unique and highly flexible combination of business development processes, infrastructure, and people designed to nurture new and small businesses by helping them to survive and grow through the difficult and vulnerable stages of early development. So we are providing nurturing, instructive and supportive environment for all students who want to be an entrepreneur, who want to be a job giver, not job seeker. The moment we postgraduate, we try to find out where I can go for a job, what is my placement. So before, maybe it's a very good thing that you are searching for your avenues, but while you are doing research, if you have something which you feel can give significant benefit to the society, significant benefit to any process, significant benefit to any product, just try it out. Just discuss with your seniors, discuss with the mentors which we have. Come to us, we will register you as an entrepreneur, and then we will ask you to make a presentation, your strategy, and then we will provide you the experts to whom you can present, and they will see whether it is a valuable dream, whether it's a dream can be further can substantiate it, further supported technically. If technically it is sound, they feel that you can go ahead, then definitely you can think about financial help, you can think about your registration help, you can think about scale up help, all those things will be provided. So in that space, in that center, we will give some space to all the students, some specific space depending upon the requirement with some minimum token registration charges. So the goal of the incubator is to increase the chance that a startup will succeed and shorten the time and reduce the cost of establishing and growing the business. So if you go on your own yourself and you try to establish your startup and business outside university, outside the center, you have to invest in space, which is very costly. If you want a space in a very posh business center or in any good locality where all the connectivity is there, where you have internet, where you have metro connection, where you have easy to, you can say, visibility. So that carries a cost. That is your first investment. Then you have to think about how I'm going to develop my product. Then you have your investment in terms of, if you are making a chemical process, investment in terms of consumables, non-consumables, your HR, you need a team who can help you. But in the university setup, you have all that available. Maybe you are a science student, but at the same time, you have department of science, where you can take you can see that your those who are in the same position in that you can make a team of a biotechnologies, team of a person from sciences, team of a person from computer sciences, and those make a company you can start. Because as a person, you cannot carry from that to the market. You need all these supporters. In my also research, all those research which I have joined hand with other partners, those research are very, very successful. Because I cannot be master of all. I can be, maybe I'm very good in pharmacy, I'm very good in formulation development, but I don't know how to file IPR. I don't know how to perform my safety studies. I don't know how to do the scalable studies. I don't know how to do the clinical startup, the clinical studies. I don't have the financial resources. But when I tied up with my colleagues, with the partners who are working in different institutes, different areas, I have done the formulation development. My formulations are evaluated by somebody who's working in an ethical setup. Then it is again critical studies were done by some other colleague who is working in the pharmacology area. So all those things happen very quickly and technically sound. So end result, my product is already, I have already transferred 14 technologies to companies as well as defense organizations. And I have seen how, because before joining Hamdard University, I was working with industry. I worked for about 6-7 years in Red Vaccine, which was now Sun Pharma, now overtaken by Sun Pharma, after Daiho. So Red Vaccine Research Lab was a research-based company where I was working in the research and development department. So how R&D department works in a company setup? We have different departments, different support departments. Our responsibility was to develop the product, and we get all the support from analytical division, regulatory division, IPR division, clinical, preclinical. Our job was to develop the product, and then rest of the studies are done by the whole team. And that's how we develop the product within the shortest period of time with highest success rate. So this is the same way you have to learn to build teams within the university or within or maybe within different universities or different institutions. In today's scenario, you have so many platforms, you have so many friends. Now, sky's the limit. You can have a friend who's sitting in USA and he can give you advice. He can make a team with you. You can have a friend who's sitting in some other university, in their university, and he can be your partner. So in this social media, I think age, can definitely make teams, get advice. You can make a team of your where you have same wavelength, we have same understanding, where you can support each other and come and have a startup. So what we are providing a shared space depending upon your requirement. It can be office space, it can be a lab space with all the internet connectivity and all those periphery which are basically required. We can provide you business advice from the experts who are known to us, who are maybe in my contact, who are maybe in the contact of Professor Hamzat Abidin, or in the contact of other schools. We have a management school, definitely they have their own mentors who can definitely give you business advice. Then services, then networking, because we have a team of mentors, and these mentors are from different fields. And we are planning to hold every month a meeting with all the mentors. And depending upon your difficulties, your problems, those mentors can always give you advice. Then we will have a full-time manager, a CEO in the center, who can definitely will be available to any on-the-spot solution or problem or support, whichever required by the startup person. So, as an incubator, what are your fears? We always think, I don't know how to, this is my idea, but I don't know how to develop into a product. I'm also fearing that if I talk to somebody, if I go to the market, how I'm going to sell this product? How much money I need? I don't have any money. How am I going to make, maybe I can make it in the lab, how I'm going to make it in the big production area? Who's going to support me? Then where I have to go for registration? How I have to apply for market registration? What are the documents, like how the documentation is going to happen? So all those things are basically our worries, because we are only expert in our area. If you are from biotechnology, you know how to do fermentation, you know how to do tissue culture, that is your expertise. If you don't know, if you are coming out, if you are making a secondary metabolite from your tissue culture, if you are making a plant like Sir has already made various fruits and plants, then how to take it to the field? He is from a government lab, he has his own teams and he has his own support system, which is getting from the government. So you are looking for support. That support is given to you with this investment and with these risks. Even if you fail, you will learn something. You will learn how to fail, and you will also learn how not to fail. I have done this thing, I have failed. Maybe next time I will not do these mistakes. And in the scenario where you have support and you have advisors or mentors, the chances of failure are much less. I'm not saying that every incubator is a success. Success depends on many factors. But success is not only financial success, success is success of your idea. Your idea is converting into product. As a scientist, I get the best satisfaction when I see, when I was working in the industry, when my product is available in the market. I can say proudly, okay, this is my product. I can show to my students, my children, that this I have developed, I was working in the industry. So it's still available in the market and people are using it. So even if you are not marketing, maybe your idea, your innovation can give rise to a product by some other company. So there are so many avenues. So our objective of starting this center is to provide focused support. So you can establish your ideas, you can make them a reality. We can support the acquisition of business knowledge from different areas. We can facilitate the raising of necessary finance through banking system, through finance advisors. We can also introduce the entrepreneurs to the various networks which we have. because now we are in an age where we have a lot of contacts and networks. And I'm not the only person who is working in the center. We have a team, and they have their own network. So depending upon your innovation, depending upon your requirement, those things can be provided to you. So we are doing all this support, and we will, as I have already told you, there will be advisory service, there will be service about finance accounting, then intellectual property. Yesterday, we have a wonderful presentation by Mr. Lalit about IPR, that we are only paper scientists, what he said. But we have to, whatever innovative idea we have, we can always apply for a patent. Mostly students are not aware that you can apply for a patent even at the time of concept. If only you have a concept and you are confident this concept can convert it into a product, you can always go for patent. You can patent your idea. You will get 1 year to generate that, and in 1 year time you have to. So initially you will give the provisional specification, provisional properties of your idea. Once in 1 year, if you develop that product, you can give the final specification, and then you will get the patent in whatever time period they have. So, because what happens sometimes, if you have something very innovative when you are doing it, maybe it can be copied by some other department, some other company. If you are discussing with your peers, it can go to. So, to protect your idea, how can you protect? By applying an early application. And your application date is your date of protection. And the most important thing, when you are writing your specification, writing your description about your invention, you have to write it in a very, very detailed manner. Because whatever is disclosed, only that is protected. If you are hiding the information, that is not protected. Because we have different types of IPR. We have trade secrets, we have patents, we have designs, we have copyright. Earlier, we used to have trade secrets. You must have seen so many companies who are carrying trade secrets. Ruabza is a very good example. It is not patented. It is a trade secret. Nobody knows it now. What is how it is being made into this product? Nobody has copied. We have been successful to copy it. Coca-Cola. Although now we have various versions of cola, but it is still a trade secret. They are not patented. But there are patents also. All of our pharma products are patented. Why they go for patenting? Why you go for patenting? Because you are getting a protection. If somebody is using your knowledge, your idea, then you can take him to court for litigation, for damages. Those things you can stop him using your idea. And this has happened. Many companies who are stealing ideas from some MNCs, and the moment they come to the market, the MNC immediately can file for litigation, and they can stop him and ask for damages. And those damages are huge. In a federal court and a US court, those damages are huge. Companies are sold out. So nobody dare to touch any innovation and invention which is patented. Because the day your application is accepted, that is your priority date. Priority date is date of your protection. From this date onwards, you are protected. Then you have legal services, office operations, all type of, you will get if you are in university, you are getting an address, you are getting an email connection. You have shared services like meeting area, audiovisual area, then your food services. Then all the management, which is basically you're running the center, they will also help you in running your own office. So these are the top few of the examples. We have innovation and entrepreneurship, IIT Mumbai, Technology Business Incubator, IIT Delhi, which is very, very successful. And IITs are more successful because they are working in an area where development process is much shorter than the area where we are working in the life sciences. Life sciences products, the life cycle is much longer as compared to if you are developing a mechanical equipment, or you are developing a software. Then we have Technopark, Technology Business, Kerala. Then we have Startup Village, Kerala, Indian Angel Network, Technopark. Then we have Center of Innovation Incubation, IIM Ahmedabad. We have IIM Bangalore, accelerators, yes, accelerator. So these are the few successful examples which are running by various institutes and universities. Now, in the incubation process, when we are going to enroll you, first you have to be eligible. You have to be a student of Jamia Hamdard in any school. Maybe you can also be alumni, and you can partner with a current student. Or you can be an entrepreneur, you can be a successful company, and you can partner with a faculty or a student in the university. So if you can make a team with the students, obviously students are going to learn, and we will definitely allow them to be part of the incubator center. Then there will be admission process in which we will ask you to fill a simple form where we will give you the detailed details of your innovation, your idea, and that will be evaluated by the team of experts. Then we will also see the intellectual property, that whatever the ideas you are giving, if it is already been reported, it's already been available in the literature, in the public domain, then those advice can be given to you that how to modify it, or maybe if it is already protected, then we will advise you not to go ahead with that. And because whatever product you are developing, it has to be innovative, first time, and it should not be something which is already existing in the literature. If it is existing, all your hard work can go to the drain, because at the time of once you go to the market, they will stop you. So it is very, very important that you do a thorough patent search, literature search. Maybe you go for a paid search. Sometimes free searches or Google searches are limited because they are not accessing the paid databases. So maybe you have to go for a paid search, which is not very costly, but you have to invest something. And after searching that, whatever idea I'm thinking about, it should not happen that it is already existing, already owned by somebody else. Because if you don't know at today, then maybe after 1 year, you will regret that I should have done a thorough search, and whatever I invested, it is going waste. So you have, we can provide you, help you by connecting with you with different banks and to seed loan. There are other government agencies who are providing various types of schemes with seed loans, like we have a technology development board by DST, we have BIREC. They have different schemes in which faculties can apply. There's a different scheme where entrepreneurs can apply. So those schemes, knowledge will be provided to you. Then you will be having common infrastructure, other support services, assessment, and information. And then we will have a tenure. Maybe the space will be given to you 1 year or 2 year, and then you will be evaluated. Once you are established, you can move out to your own premises, your own factory. So, and then conflict of interest and all those regulatory things and agreements. So, okay, just two slides more. Yeah, I'm just finishing. So, conflict of interest, basically, wherever you have a team and partner, there should not be any conflict of interest with respect to your technology, with respect to your regulatory and IPR things. So, in Jamia Hamdard, in 2017-18, we have established Institutional Innovation Council, which is running since 2018. It was already registered with the AICT and other bodies. Now, in 23rd May, we have this Center of Innovation, Incubation, Entrepreneurship in our nanotechnology building on the second floor. The space is given to us. It is inaugurated by Dr. Subhash Sarkar. There are a few images, and there are many student innovators who exhibited their innovations and some of the startups. Our vision was to create, guide, and embrace the student entrepreneurs in order to build successful business that are sustainable and productive. And to achieve this vision, we have a mission that we will nurture the youth, and we will provide. In the wrong direction. So we will provide mentoring, developing them into self-reliant entrepreneurs and turning their ideas into reality through access to professional mentors, collaborative office spaces. I have already said what we are doing. These are the main activities. And this is the USP of our startup, where we are doing through innovations. So this is how we are working. You have an idea with inspirations, opportunity, and mentorships. It can be gone to the community, you build the enterprise, and it is a hub where you have all these things are finding it together. Then, so this is the features which I already discussed. So our process is listen, brainstorm, design, develop, test, submit, and marketing. This will follow. So we have various incubators. So in the end, other famous quote from very successful Mr. Narayan Murthy, I quote, if we wanted to create a great company, we should be Optimistic means we should be positive and positive about our own idea about success. Another quote by Azim Premji, who is the chairman of FIPRO: if people are not laughing at your goals are too small. So even if they're making fun of you, even if they're criticizing you, take that criticism positively. Use that criticism to build your product free from defect, free from problems. So with this, I thank the organizing committee for giving me this opportunity. So in early stage startup, success is not a to-do list, but it is a to-learn list. Thank you.
Thank you very much for your excellent talk. Because they have big what is part of the traditional case. So it's a right way to get plans of what we can do.
Now I would request Dr. Pawan Jadoo to please felicitate Professor Paranjali Sena, sir. Thank you, sir. Now I request our next thought-provoking and visionary speaker, Dr. M.K. Reddy, sir, to please grace the stage. Dr. Reddy is the group leader of Crop Improvement Group, International Center for Genetic Engineering and Biotechnology. He is working on the development of herbicide-tolerant crop plants for weed-free agriculture and also targeted genome editing for improving the agronomic performance of crop plants. His research group developed rice plants simultaneously resistant to more than one herbicide with different mode of actions to control the weeds without causing any injury to the rice crop, and implementation of herbicide-based weed management in rice cultivation. His research team also developed transgenic technology to enhance phosphate and nitrogen use efficiency rice crop. Recently, his group extensively used CRISPR-Cas technology for targeted genome editing in rice and developed several edited rice loci that improved the architecture of rice plants to enhance the overall rice productivity and also adaptation to environmental stresses. I request Dr. Reddy sir to please address the audience on the topic targeted genome editing for improved agronomic performances in rice.
Good morning to all of you. I thank organizing committee, particularly the Professor Abdin, for giving me an opportunity to discuss some of our research achievements in this conference. The topic of the topic of my talk is targeted genome editing for a improved agronomic performance in rice. During my talk, I will cover increasing the rice productivity and also the sustainability of rice productivity. The increasing rice productivity by increasing the number of panicle bearing tillers or the number of grains per panicle or the grain size and the weight, so that we can increase the productivity of the rice. The sustainability of the rice productivity is by reducing the crop losses by abiotic and biotic stresses. So we can develop a abiotic stress tolerant rice plants or the biotic stress rice plants. We can make the sustainability of the rice product. In a classical breeding, generally we require the availability of a natural genetic variability or a beneficial alleles in the population. So is a prerequisite for introgression and developing a improved variety. But a lot of scientific research efforts are in the rice community identified a large number of mutant or the beneficial alleles and their association for the rice improvement. But these beneficial alleles are widely dispersed. One beneficial allele will be in some variety, another will be some another variety. So the availability of these mutants to the plant breeder is a bottleneck, and the introgressing these beneficial alleles from a wild relatives and other things into a cultivated variety requires a lot of backcrossing and breeding cycles required. So with the targeted genome editing technology, it is become easy to generate that genetic variability within the cultivated rice itself. We don't require actual mutants. We don't require time-consuming breeding cycles and the other selection processes. So with the one can edit any genome by expressing the Cas9 and sgRNA. This Cas9 and sgRNA technology is the recent addition to the genome editing technology. The Cas9 is a naturally encoded protein with a 2 DNA breaking domains. So this Cas9 is a endonuclease, but it cannot break the DNA its own. When you give the sgRNA, which is interacting with the Cas9, and the sgRNA will guide the Cas9 to the desired place by the investigator, and there only the specific place the Cas9 will make the double-strand DNA. This double-strand DNA break will be repaired. While repairing the things, we can edit the things what you want to edit the things. In our laboratory, we have developed a large number of edited rice lines. During my talk, I will give you a few examples, whatever the time permits, how we can use this technology to improve the rice productivity or the sustainability of the rice productivity. Suppose in this particular case, the YSTB1 gene, when it is expresses, it suppresses the tiller number of the rice. When this gene is expressing, the tiller number will be reduced. So we want to target the things to knock out this gene so that we can increase the tiller number. So to do this, we have first we put the Cas9 and sgRNA and cloned into the plant transformation vector and develop a first transgenic plant which is expressing the Cas9 and sgRNA. When designing the sgRNA, we use some software which is unique to this gene in the rice genome, so that the Cas9 will be guided to this genome, this region for a double-strand break to knock out this gene. When you have done this thing, then we have taken the
and please forward and
when we so that I don't get the pointer here properly.
this is the first button. Yes, sir. Yes, that's right.
After making the transgenic plants, we want to see that plants are edited in this area or not. So we have designed two primers to amplify this region from the transgenic plants. and sequence this area. For the PCR product was sent for a sequencing. When you sent the PCR product for sequencing, this is the place I am targeting my Cas9 to make a double-strand DNA break. When I send the sequencing PCR fragment for sequencing, the sequencing results were very clean up to this area where the editing is happened, because we have two alleles for this gene from the male side and the female side. Both can be edited or any one can be edited. So in this particular case, one allele was edited, one allele was not edited. That's why the sequencing file has a overlapping sequences from this break onwards. Because one allele edited, one allele is not edited, so you can have overlapping peaks in that area. So that when you see the next thing, so you can find by selfing those plants, you can say make the homozygous or biallelic editing. So in this particular case, I have deleted 24 base pairs from this region. When I put this sequence into the thing, this is deleted 24 bases, but the frame is restored back in the translation. This gene is not a truncated or a knocked out, but only deleted 24 bases. Then we found another mutant with a 13 base pairs. 13 base pairs deleted here. Now the frame shifted, so the protein is truncated and the gene is knocked out. When you see these plants, this is the edited plant which gives the 38 tillers and gives the 38 spikes. The original plant gives the 13 tillers and the 13 spikes. By editing one gene, you can see the yield from this place to this place. How much double, triple the yield in the rice. Because you increase the number of tillers from 13 to 38. All the 38 tillers are synchronized and flowered and given the spikes, and the yield is increased heavily. So this is the power of the genome editing technology without using the breeding and other things. So I will show you another example. This is called a dense erect panicle. In this dense erect panicle, this gene, when expresses, the spike of the rice is very loose and less number of grains are there. When you knock out this gene, the spike is compact and more grains in the spike. So when you take this, so we want to knock out this gene to increase the grain number in the spike. So taken these target sequences and then transform them, we develop the knockout line. So there is a one base inserted at the broken double-strand DNA break, and because of one base insertion, the reading frame changed and the gene is not functional. So because of that, you can see the increase in the grain number per spike. So when the normal spike is there, compared to this edited spike, a compact spike and with the more grains, and the total yields increases using this technology. So another example for this increasing the yield is the cytokinin oxidase gene. What the cytokinin oxidase does, it will oxidize the cytokinin and make the cytokinin inactive. The cytokinin is a very important enzyme, hormone for the plant, for the cell division and the cell elongations. When this particular isoform, cytokinin oxidase, expresses in the developing spike of the rice. When this gene is expressed, it is inactivated the cytokinin, the spike becomes smaller. So less number of grains. When you knock out this cytokinin oxidase, then more active cytokinin will be available. So the spike become a bigger and a larger number of seeds will be there. So we have taken this cytokinin oxidase gene and knocked out, and there are two bases deleted in this knocked out mutant, and the gene is not functional anymore. So you can see the edited lines has a bigger spikes with more grains compared to the original. Now you can increase the grain size also. Grain size and this GWS2 allele is a ubiquitin ligase. This ubiquitin ligase will inactivate the expansion gene. Expansion gene expressing in the rice husk. When the expansion gene was knocked out, the husk will be smaller. So when the husk is smaller, the smaller grains will form. When I knock out this ubiquitin ligase, then it is not inactivating the expansion gene in the rice. So when the expansion gene is active, the bigger husk will be formed and the bigger grains were formed. So that's what happened. We have knocked out this gene by inserting a one base into the edited line. Then you can see the bold grains and the higher yield in these things. In this example, we have knocked out the phytic acid pathway. The phytic acid is an anti-nutrient. It is there in all cereal grains and legume seeds also. Because the phytic acid is a 6 phosphates in an inositol ring with a negative charge. All phosphates have the 2 negative charges. There are 6 phosphates. There are the 12 negative charges in this molecule. This phytic acid is not digested by our system. So this heavily negatively charged molecule will chelate all your minerals, and you will all the time you have a mineral deficiency. Because the bioavailability is there, the availability of minerals in our food is enough. But this is chelating out, the bioavailability is the problem because of this anti-nutrients. So we have used this CRISPR technology to knock out this phytic acid pathway at the end, because the other intermediates are very important for the plant development. But we have knocked out this gene from at the end so that we can have a phytic acid free rice grains in the plant. So now I have shown how to increase the rice yield. So now I will see how to protect from the reducing the yield losses through abiotic and biotic stresses. This particular gene, what is called rice ERF109 isoform, increases the abiotic stress tolerance by knocking out this gene. So my student Himangani has chosen this gene and developed a knockout line. In addition to the abiotic stress, she knocked out this gene. There are 11 bases were deleted and the frame was shifted and the gene is non-functional. Because of that, protein reading frame was changed. So this plant will protect from the brown plant hofer. The plant hofer is a major pest in the rice. It will infect, it completely burns, dries up sucks up all the things. When the edited plants was not eaten by destroyed by the hofer, the control plants was completely destroyed. Similarly, another student working on the biotic stress, particularly the blast resistance fungus in the rice gene. When a biotrophic pathogen infects the plant, it will start feeding on the living tissue. So to survive, to feed on the living tissue, it has to suppress the innate immunity of the plant. As soon as the biotrophic pathogen infects, it will activate some of the genes from the plant side, which can suppress the innate immunity of the plant, so that the biotrophic pathogen can able to survive on the living tissue by feeding. So those genes which are induced by the pathogen are called as susceptible genes, because when they infected and express the susceptible genes, so that the environment for their biotrophic mode of life is easy. So my student has taken those genes to knock out the susceptible genes, so that the plant and the pathogen cannot induce them to suppress the innate immunity. So the plant innate immunity automatically protects the plant from this. she has chosen a set of genes. These are all proved in the literature by using a RNAi technology. By doing this, the innate immunity increases. So my student taken that genes and try to knock out those genes and make the plant innate immunity, improve the thing so that all the biotrophic pathogens can be protected from these things. So she has done a phospholipase D1 gene inactivated. Now she is working on the how this protects from the fungus. So now I coming back to the abiotic stress. You see here the trehalose is a very important disaccharide. Generally, the sucrose and the trehalose are the two disaccharides exist in these things. In the evolution, the sucrose was taken over in the most of the land plants. The trehalose was taken by the lower plants, so pteridophytes and other places to make the things. But the trehalose has many advantages for protecting from the abiotic stress. Extreme abiotic stress, it will protect. What happens when the trehalose was available, when the dehydrated or the temperature is open, it become a glass. It freezes into a glass. Before the cell dies, it become the cells are frozen in the glass kind of material. So the cells are still alive, but they are dormant. When you put the water back again, the glass melts and the cells start working, so that they never die, resurrection kind of things. But that kind of technology they want to develop by using the two genes for that halosynthesis, but not successful in the literature because a moderate tolerance came. The reason for that is there is an enzyme called a trehalase in the expressing in this thing that is degrading the trehalase into glucose. Because when you overexpress these things, expressing, I will increase the trehalose and make the plant resurgent plant. But this trehalose is not accumulating that high because the trehalase enzyme is degrading it back. So we use this technology to knock out this trehalase. Then we have knocked out this trehalase. We have deleted almost 363 bases from this gene during the editing process, and we developed the trehalose knockout mutants. These mutants will tolerate the high dehydration stress compared to these things. But actually, what happened? These trehalose biosynthesis genes are existing in the rice, but now they are actually differentially expressing. The pollen grains, where the desiccation has the problem, there the trehalose will protect, and the desiccating embryo in the seed, where the extreme desiccation, but that's the living tissue, but it has to be protected. There the trehalose will be there, but it is not constitutively expressing. Now we have developed a knockout mutant for trehalase. In that knockout trehalase mutant background, we want to express these genes so that our rice can never die. Like that, that's our hypothesis. We are in the process of overexpressing these genes into the knockout mutant so that high trehalose accumulation can be developed so that the rice plant can protect from the extreme temperatures and also the desiccation. So this is another example for developing a drought tolerance. My student will make a presentation after me. She will explain the more details on this technology, how to develop the drought tolerant plant using another concept. So I will stop it here. So I will take any questions.
Question one is, so the question is, what type of cell type that
No. These are all the agrobacterial mediated transformation and the stable lines was developed. These are heritable and these are permanent, not a cell lines. Yeah.
So the question is, genetically modified, have a problem with communication. So what kind of ability thing can happen so that for product can be
So recently, the government of India is deregulated the edited plants if I remove the Cas9 and other things. Because whatever the Cas9 and guide RNA components I put in the plant, that is going to integrate somewhere randomly in the genome. But it is integrated in one place, editing some other place. So these are not linked. In the next generation, I segregate out. Now most of my plants are mutants, not a transgenic anymore. So these are deregulated. Anybody can use it. If I say these are my edited plants, I'm telling. Otherwise, I don't tell this is a mutant. The natural mutant also happens like this. Because most of these my examples I have picked up from the Japonica rice. These mutants are available in China and Japan and are extensively used. But in Indica, these mutants are not available because I have chosen all those things in prior information and created all the mutants in Indica so that my success rate is very high because these natural mutants are successfully used there somewhere else. So that's why I can create here. That's why the things, otherwise you can, with your knowledge, you can plan the things which gene to knock out, which gene to pathway to control. That's a different story. But all my examples are existing before in a natural way, but I created those things in my particular rice cultivar so that I don't require any breeding and other things, actual mutation or the breeding. So I can create all those things. Huh? Generally, because all these genes were not mutated in one variety. I can pyramid now because all are in the same. Each one was separately edited because in the genetics, the genes are the pleiotropic effects will be there, so the interactions will be there. But all these examples I have chosen, these are well established for their positive effect, not the negative effect in the nature, so that I don't have seen any negative effect. No problem, sir. You can ask.
That you need to design, which we talked about. So is there any other in the market apart from that? Because that will interfere the whole trials.
So generally, the people says in literature, there is a competition for a sucrose and a trehalose. So all the higher plants are adapted to the sucrose. Now you are increasing the trehalose as a byproduct like this, but the amount of sucrose is made as enormous. It is the starting point for many things. It's not a limiting thing. By increasing my trehalose is not really a bottleneck for the sucrose, limitation for the sucrose. So I don't expect anything in there. So by presence of this disaccharide, it's like another sugar. So it will protect the osmoticum like thing, or also protecting from the many other things. These are well known in the animal side and other things. Some of the animals which go for a long what is the winter sleeping positions or the other desiccation places. These are the things which protects them. That time they accumulate. This is known in the biological system. Now I am imitating that to introduce into the plant. Suppose you take a pteridophyte salaginella, you take that out, dry. It's completely dried. You sprinkle some water, tomorrow this live. So a similar thing can be produced in the rice.
Thank you, sir. Now I would request the chairperson to please felicitate Dr. M. K. Reddy with a memento. Thank you, sir. The next distinguished to honor us with his presence is Dr. Niranjan Chakraborty, sir. Dr. Chakravarti is the Professor of Eminence in National Institute of Plant Genomic Research, New Delhi. He did his PhD from JNU. He had many research papers in reputed journals and a number of book chapters. 36 US and Indian and PCT patents. Dr. Chakravarti's area of interest is stress proteomics, gene expression and regulation in crop plants. He is the fellow of Indian National Science Academy, National Academy of Agricultural Sciences, and National Academy of Sciences, India. He has received various awards such as DBT Award of Biotechnology, Overseas Association, and ICCR Commonwealth Scholarship, and many more. Now I would request sir to please enlighten us with his talk.
This is forward. This is pointer. This is pointer. This is forward. Backward and only forward and pointer. The chair and the audience, very good morning. The topic is plant organelle or organellar proteomics, turning knowledge into application. So don't think about application because the presentation I will share with you is simply a molecular biology topic, and for plant molecular biologists as well as animal molecular biologists will understand this. Understanding stress tolerance. Spatial partitioning of cellular processes is a fundamental feature of life, which facilitates multiple parallel pathways to occur in specialized subunit or the organelles. Therefore, the knowledge of organelle proteome is of paramount biological importance and is critical to understanding stress tolerance or disease resistance. It is same for plant as well as for animals. Now the question is, why proteomics? Why not genomes? All of you know that genomics, all these terminologies came in 1986. And exactly after 10 years, we found new terminologies. Protein, proteome, not protein, proteome and proteomics. So proteome is nothing but the total content of protein. It can be in a cell, in tissue, in organ, in whole plant or animal system. 2000 is very crucial year in biological science because you all of you know that this was the year when we got draft sequence of human being, Arabidopsis and rice and so on. So there was an interesting paper in Science that year, the year 2000. It is a comparative genome. And you can see they have taken, the authors taken 4 different organisms. The first one is a bacteria, Haemophilus influenza. Second is Saccharomyces cerevisiae, C. elegans, and Drosophila. And now let us compare the genome and the complexity of the organism. 2000 gene is there in a bacterial cell, which is Haemophilus influenza. It's a very simple structure. And the second one is Saccharomyces cerevisiae. It is more complex. So there is a correlation. The number of gene increases, the complexity increases. It is true. But you are confused, go to the next 2 columns. C. elegans, it is a ribbon-like structure, very simple structure, but the number of gene is 20,000, 18,000 roughly say. And then Drosophila, which is more complex an organism, which is everything, eyes, wing, leg, and entire body. However, the gene number is 13,000. So the question is, what is the problem here? Because the genome mass is not related to complexity. That means if you know the gene or genome of an organism, you do not understand many things, the regulation and many other complexity in the organism. There comes the proteomics. Now, as I said, that 1996, first we came to know proteome and proteomics. Proteomics is a study of proteome. So that was the year that my laboratory started proteomic work. And we have taken a different way. But first we studied wholesale level proteomics in plant and plant two different plant. One is legume, that is chickpea. The other is the cereal, which is rice. So what we did actually, fractionated the entire cell and then taken out one by one the membrane, the nucleus, the cytoplasm, the cell wall, the chloroplast, secretome, mitochondria, every part we isolated independently and studied the proteome landscape of that organelle to understand that what these proteins are actually doing in the organism. Has they any role in stress tolerance or disease resistance? So I have a relationship with this university, incidentally, and during the Corona time, we actually were studying plant cytoskeleton, and if you go to literature, you will not find cytoskeleton proteome either in plant. Plant is absolutely missing. In animal, few paper you will get. And there was a MSc student from this university who joined in my lab and the dissertation work. And during Corona time, the entire system was collapsed. He worked few months with me, and then we made a paper out of it. And just September, it published in Plant Cell and Environment. Now it is online. You can see. His name is Hadi. I don't know whether he is in the audience or not. So this is the relationship of. I will tell some more. So in proteomic studies, I will share with you two different case studies and see how it is important to understand stress tolerance in plants. A platform we developed long time ago, and this is the plant I mentioned, chickpea. So what we did, many commercial chickpea cultivars we put into stress, particularly water deficit stress or dehydration. As you can see here, unstressed condition, the leaflets are flattened. When it goes to dehydration, you see curling of leaves and ultimately wilting of seedlings. At this position, you can re-water the plants and wait for 24 hours. Here it is R24. That means you give a choice to the plant to recover, and you can see the recovered plant get back to the normal. And this is the case you will get if the plant is resistant. If the plant is susceptible or hypersensitive, then you will not get back these plants to this. So this is the starting material. Then isolated those samples and fractionated the way I showed in the previous slide. So the first organelle in a plant, obviously the cell wall. So our first choice was to study the cell wall. Here again, Jamia Hamdar student Shaista Parvin, that time she was doing PhD. So she started this work. As you can see, that these are the isolated cell wall. And if you take section of the cell, then you can stain with PTA. This is the middle lamella of cell wall. This is one vacuole. This is another vacuole of the adjacent cell. And you can see this colored line is the plasma membrane. That means if you isolate cell wall, then always it will be accompanied with plasma membrane. You purify it, and here you can see. Can I have any other in? No, this fine. So here you can see the purified cell wall without any plasma membrane. That can be further tested with enzymatic analysis, because if you study enzyme, then plasma membrane associated ATPase will be there. You can see here the crude fraction is having ATPase activity. Then the purified, you will hardly get any activity. So you understand that this is a purified cell wall. Next, we developed the proteome. That time my lab was doing only the two-dimensional approach of doing proteomics. So here is the proteome, total cell wall proteome of chickpea. And when we functionally annotated, we understood that about 22%, roughly 20% proteins are associated with stress tolerance or disease resistance. We did many other bioinformatic analysis, but I would like to emphasize on this. We got 4 this red and circled spots you see. These spots were highly upregulated protein. We did ask, what is this protein? And appeared to be all these spots were ferritin. Now the confusion is that ferritin, any plant scientist would know that ferritin is a protein that resides in the chloroplast. How did you get in cell wall? Is it an artifact or it is a new discovery? So we took this challenge and then studied it and functionally annotated. So the protein was ferritin. We named it CA ferritin because the scientific name of chickpea is Caesar arietinum and ferritin is ferritin. So the name is CA-FR1. It is a small protein, 20 kDa protein. When it functionally annotated and understood clearly that it is a new ferritin other than the chloroplast protein, so we filed a patent, and luckily we got a US patent on this protein, CA-FR1. It is the first discovery in plant that ferritin can also be a cell wall protein other than chloroplast protein. So we cloned it and then studied deregulation of this gene. As you can see here clearly, when you put pressure like dehydration, it is upregulated. When you put into hypersalinity, it is upregulated, and it is also upregulated when you inflicted plant with ABA treatment. That clearly shows that the upregulation of this gene is associated with ABA-dependent pathway. This is clear. Next, so we took a heterologous system to preliminarily study its function, and the system was Saccharomyces cerevisiae. So what we did, we have taken a mutant. So now see, so this is the wild type. Then we have taken a mutant, we complemented it, and also overexpressed. Now you put this strain into stress condition, and we found it, to summarize it, that when it is overexpressed, it is more resistant, and the complemented lines also resistant. So it gives an indication that it does participate in stress tolerance. And you can see here the overexpression of ferritin in the complemented lines as well as in the overexpressed lines. So then we thought that we should study the functional annotation also in the plant background. So we have taken Arabidopsis, and the gene function can be done in 2 different ways. One is minus way, that means you make a mutant, or plus way, you overexpress it. So the first thing we could do, that we downregulated this gene in Arabidopsis. As you can see here, this is wild type Arabidopsis. The plants can grow, and even under stress condition, also it can germinate and can grow. But if you mutate ferritin, then you can see even the plants in the plate, it cannot even germinate. That means ferritin is an important constituent of plant, which is not simply a iron storage protein, but it also help in stress tolerance. Because when you downregulate it, it actually loses its ability to grow. We have studied also other biochemical analysis, which I am not going to give you details. So till now we understood that ferritin can also be in plant in the cell wall, and which actively participate in plant stress tolerance. Finally, we overexpressed it. First was a mutation, and now it overexpressed it. And as you can see here, two different lines we have taken, overexpress 4, overexpress 3, and compared with wild type, you can easily see these plants are early flowering plants. And only one emphasis I will give here, that we studied the ROS pathway in those plants, and by NBT and DAP staining, you can see the radical, superoxide radical and hydrogen peroxide production was much less in the plant. That is one indication that probably it works in tackling oxyradicals and give plant support to sustain stress condition. The next question is, is it possible that one polypeptide or one protein gives so much tolerance to plant? The answer could be no. So it must be having some associated protein. So through string operation, we understood that several genes are associated with ferritin to give plant strength to sustain the stress condition. And one of these important candidate was IRT1. So this we got from string operation. And then we did co-immunoprecipitation and reverse co-immunoprecipitation and proved clearly that ferritin actually associated with IRT1 and gives plants protection against abiotic stress condition. IRT1 is also an iron regulatory pathway gene, so it makes sense. So the conclusion is, we discovered through proteomic analysis that ferritin can be also present in the cell wall. It interacts with IRT1 and probably gives plant strength to survive under stress condition. This was case study 1. I will take another couple of minutes. Okay. So the second study was with endomembrane, and the protein is SUN1 protein. It is also from chickpea. So the name we had given, Caesar Ariatinum SUN1 protein. So what is SUN1? Dinesh has done this work. The first work was done by Shaista Purbin, and this was done by Dinesh Jaiswal. So similar way, we have isolated endomembrane from plant, purified endomembrane, and then we could detect the protein in nuclear inner envelope. After sometimes I will show you that how could we understood that the protein SUN1 resides in the inner nuclear envelope. So the similar way, we have seen across the dehydration type points how the protein behaves. Ferritin was continuously upregulated protein, but SUN1 is a protein which was downregulated. Whether it is upregulated or downregulated, the proteins which are deregulated by stress, we call them stress-related protein or stress-associated protein. The SUN1 is important because it depends on two domains in the protein. One is UNC and then SERB1. It is a linker protein. Linker protein because it links between nucleoskeleton with cytoskeleton. So it is having a very important function in any organism. It can be in animal system as well. So based on these two domains, we named it SUN protein, and since it is from Caesar Ariatinum, so the protein is SUN1. We localized it. As you can see here, the SUN1 YFP protein is in the nucleus, but we cannot tell that it is in the inner envelope. We also stained with DAPI and then merged it, and you can see clearly that YFP merged with DAPI. So we clearly could demonstrate that SUN1 is a protein localized to the nucleus. This is transiently expressed. But when we make transgenic, stably transformed organism, here you can see the SUN1 YFP in the inner envelope, and interestingly, it was also expressed in endoplasmic reticulum. That gives an indication that it is a linker protein indeed, because since its association with nucleoskeleton as well as with cytoskeleton, it expresses in both the cases. And we also found from literature that some protein also take part in stress tolerance, and that is UPR, unfolded protein tolerance. To which we thought that we will also functionally annotate it. And this protein is a little bigger protein, 68 kDa protein, which transcript size is 2 kb. Again, we filed a patent, and we got a US patent on this protein. So the protein CS1 is also a protected protein. Now we see how we did functional annotation of this protein. So similar way like ferritin, we also studied that how it behaves under stress condition. And as you can see here, it is dehydration. On stress condition, the expression is this, but with stress condition, it is deregulated, particularly downregulated. This also downregulated under sodium chloride, that means hypersalinity. And incidentally, it does not do much in ABA pathway. So we understood that this is a protein, SUN1, as against ferritin 1. This probably was independent way other than ABA dependent pathway. And we also studied its expression, tissue specific expression, and you can see here that it expresses almost every organ from root to flower, although the expression is more in flower bud and flower. So we concluded that this protein, this stress responsive protein, is deregulated by stress condition, but it must be having some function in plants development. So the similar way, we overexpressed and complemented in Saccharomyces cerevisiae, and here you can see that when you complement it or overexpress it, the stress tolerance level increases, although it is not exactly the wild type condition, but almost 50% stress it can relieve. And finally, we overexpress again in Arabidopsis. Here you can see this is overexpressed line, this is also overexpressed line. We have taken a mutant from Arabidopsis, and this is wild type. And you can see under different stress conditions like oxidative stress triggered by methyl bialogen, DDT, and ABA, these plants are resistant. That means the overexpressed plants are resistant, while the mutant and wild type plants are susceptible. And we kept on growing those plants, as you can see here, overexpressed 1, and this is the complementation in Arabidopsis SUN3, and you can see that the fresh weight of the plant is much more in overexpressed plants when it compared to the wild type plants. So in conclusion, through proteomic analysis, you can identify new proteins, new candidates which are associated with stress tolerance. And finally, if the functional annotation shows that these proteins indeed give strength to the plant, it can be taken to the next level of biological research. That means you can make transgenic. Now both the ferritin and SUN1 protein, we are introducing into the native background, that means chickpea, and to study that exactly what we saw in heterologous system or in Arabidopsis, can it be replicated in its native systems or not. Thank you very much for your kind attention.
I have one regarding
You
you're talking about protein interaction.
I think I mentioned that to understand the mechanism any protein function, actually the student what they do to make life easy, first they go for bioinformatic analysis. And I mentioned that string analysis you can have. So you will have a preliminary knowledge. This protein must be, associated with ABCD of many genes. And then through co-immunoprecipitation, when you get the spots and do mass spec analysis, that time you do not know whether these proteins are transcription factor or any functional protein. But if you have interest, you can more study and find out that it is not unusual that the protein you will get as an interacting partner, you cannot trap transcriptional regulatory protein. There is possibility that you can get a transcription factor. I would have loved to take any question from students. I was expecting that to ask because the topic I have taken is a proteomics and not new field. It is almost 2 decades. Yeah, please go.
First student.
No, you can. You can.
Wonderful presentation. And is there any structural or functional differences in the cell wall ferritin as well as the
Oh, it's a great question, but I tell you, in studying ferritin, I could not go to that level. The difference between the structural and functional aspect of cell wall ferritin and the chloroplast ferritin. But a little knowledge I have why this protein is in the cell wall, not in the usual compartment that is the chloroplast. I found its N-terminal region is having 5 serine residues. And I was looking in literature that is there any function for this serine residues for its localization to the cell wall. I found that there are some reports that the protein in their N-terminal position, they have multiple of serine residues. They tend to have go into the cell wall. Another note that now because I have seen many proteins, dozens of proteins in different organelles, now I know that there are many proteins. The protein you may find in the cytoplasm and the same protein you can get into nucleus. Not necessarily that is a transcription factor. And we always term these proteins are a migratory protein or trafficking protein. Sometimes it resides in the cytoplasm, perform a particular function, and sometimes you find this protein into nucleus doing some other function. So multiple places a protein can present, and in multiple places, not necessarily that it exerts the same function, it can give you multiple of function. This is the beauty of protein into different organs. You do not have any, either you understood very much or you are. Thank you very much.
Thank you so much, sir. This was really a very wonderful talk. We are delighted with your presence. Now I will request the chairperson to please felicitate sir with a moment. Now, sir, I would request Mr. MZ Darby to please felicitate the chairperson with a memento. Thank you so much. So this was the end of the plenary sessions. Now we will be resuming here after 10 minutes for the technical sessions, which will be run parallelly in both the halls. So time for the coffee break now. Thank you all. अच्छा आ गया आपने so much.
इनका पेन ड्राइव वगैरह लेना है जी। कितने कितने प्रोफेसर लेक्चर्स करने हैं टेक्निकल सेक्शन?
They will not be building this. Sure, I need to change the title also. Sure, एक बार बाहर जाके सुहेल सर को कॉल कर दो कि सर 10 मिनट्स में सेशन स्टार्ट होगा हॉल 2 में. सुहेल सर, श्योर सर, प्लीज थैंक यू. उसमें ये अपना मेक कर सकते मैम जामिया हमदर्द लिखा होगा उसमें कहीं अब उसके बाद ही चीजें करें.
विंडम का
ये पेन ड्राइव में यूज नहीं सकते ऐसे. If you want, we can delete it after. अच्छा अच्छा, no problem. Yeah, open it so that I can change my title if not change it.
हेलो मैम,
Hello, sir.
हाउ आर यू? और क्या हाल है? ठीक है।
So, आज डायरेक्टर साहब का presentation होगा.
अच्छा
उसका क्या करें?
अगर जो जैसे आपको बहुत अर्जेंट है जाना है तो चले जाइए, फिर जो है और नहीं अर्जेंट है तो जाए. हाँ, देखिए आप रह सकती हैं तो ठीक है. कहाँ पे हैं? हाँ,
हम लेक्चर चेयरपर्सन मैं टाइम देख रही हूँ.
देख लो.
खत्म हो जाएगा.
कोई नया डायरेक्टर आ गया SERB का?
रमेश सोंटी जी आए.
अच्छा अच्छा, रमेश.
सोंटी जो NIP से आए थे.
अच्छा अच्छा.
मैम जो माइंड अगर पवन से पहले एक लेक्चर देने के लिए मैंने रिक्वेस्ट किया था टू डिलीवर हियर बट हमारे पहले सेशंस रैन हो गए. मुझे थैंक यू सो मच. मैम आप चाय ले लीजिए, मैं फिर दूँ. ये मुझे बस ये हाँ मैं ये डाल देती हूँ. रिविजिटिंग प्लीज हैव टी. ये टाइटल पर श्योर श्योर I'll pick that from you. Sure, ma'am.
नहीं मैं देख रही हूँ डिस्कस किया यार ये चल क्यों नहीं रहा है ये लैपटॉप मुझसे कमी थी लोगों की प्रेजेंटेशंस पे बनाओ प्रेजेंटेशन का performance मेरी भी कॉपी करना तुम्हारा नेक्स्ट में है क्या ओरल में है खुद से भी मैं ना वो दिखा रहा है एम2 वेरी गुड फिर सुबह मैंने दोबारा कॉपी किया है मेल पे है हाँ मेल पे क्यों ले रहा है ये लिखा है ना कुछ इसको पता है इसने कॉल कर दिया हमको छोड़ सकते पता है
Yes, yes, because you finished very early. Yeah, nice. Yeah. Is there any C3 and C4 alpha is also running? No. Presently, we don't categorize C3 and C4. Especially, we consider these marine springs as more C4 because the CCM mechanism is present in both freshwater and water. But CCM mechanism is more active in marine springs rather than freshwater. So, so that's the reason we are trying to differentiate how there is a difference between CCM mechanism of freshwater and marine. But it's very active in the marine springs because of low levels of carbon dioxide in the oceans. But as part of higher plants, there are very well designated. Yes. For lower plants, maybe not. Yes.
So now our next invited speaker is Dr. Ajay Arora. He is an eminent principal scientist at ARI. His area of specialization includes plant physiology, genetic engineering, and functional genomics. His one of the most important contributions is the development of transgenic tomato with longer shelf life to avoid post-harvest losses.
Thank you. Thank you.
This is forward.
Yeah.
This is pointer.
So, okay, good afternoon, everybody. Say good afternoon. Reason being, thank you. Reason being, why I am saying so, our chairs are very, very strict with the timings. I know that. And I really feel pity for Dr. Tanushri. She was having a wonderful work, but could not able to. Frankly speaking, we scientist people are really hungry for these kind of audience. Because at our home, nobody listened to us. Neither children nor wife. Wife, everybody. So please bear with me for another 18 to 19 minutes. I will try to finish faster. Sure about that. So the topic is stay green, staying alive or going to die during terminal senescence. I think little tricky. Most of this, I think the hall is full of young people. I think 95% are students. So young, I am using the word, reason being, the young population is good to stand against any kind of stress. This is my feeling, because I also passed through that stage. But we old people, or going to be old, are really fragile in nature. Okay, so that's the reason we hypothesized that stay green will do certain kind of miracle. So resilient against any kind of abiotic or maybe biotic stresses. We are working with the abiotic stresses. I know since morning 9 o'clock, you people must be hearing so many lectures regarding abiotic stresses, right? But I'll tell you the genesis. The genesis, basically, I got it from the sorghum story, and I thought that story is really useful to avoid the abiotic stresses or to delay the process of senescence. If we can delay the process of senescence through stay green trade, I think that will be really useful for the plant system also. So this is my sincere advice in between to the youngsters, that whenever you want to do certain kind of good research, you should read some of the mammalian science research or animal science research. Because most of the plant science research, we got the clue or we got the idea from these sciences. So this is for your advice whenever you read some of the papers. So I think you should go from there. So, but in functional stay greens. So functional stay greens, if I'll use the word, that means they should be highly photosynthetic in nature. And More important, the functional staggering trait is associated with the transition from the carbon and capture to the nitrogen mobilization phase. So there are two terminology I'm going to use. One is staggering, another is stem reserve mobilization, SRM. So these two terminology will be opposing effect. Okay, one is counteracting the other. So if staggering will be there, the mobilization will be less. So I'll come to that in detail, little detail, slightly later. So maybe I think that's the reason. Maybe if you can blink your eyes and take this photograph in your eyes, you will better understand the future story, right? These are some of the hormones, because I'm just introducing my talk, right? So these are some of the, okay, these are some of the hormones which plays a key role during the plant senescence. I know I'm talking about basic sense, but I think in a very, very simple language, I should translate what we people are doing. And if you can have any kind of query, we can interact later on. So these are some of the hormones where the cytokinin is going down, and it is playing really a wonderful role in collaboration with the senescence process. So that's what we are interested in, this particular hormone. And another one is ethylene. So these two are going from senescence to the death. So you can understand the importance of this. But today I will talk only about cytokine, not ethylene, because that is some other day story. We'll talk about that. So this is the hormone signaling pathway where a trigger of cascade of events involving receptor molecules, transcription factor, kinases, and small r. So a combination of these four major factors will involve in the senescence process. We will take it up for just for introduction. So cytokinin has a strong anti-senescence effect. Everybody knows it. Since class 11th onwards, we are reading this. We have a lot of evidences with us. And when we started our experiment with this particular gene, IPT, isopentenyl transferase, this is substrate inducible enzyme, right? And opposing of this is the CKX, which is degradation of cytokinin, right? CKX is cytokinin oxidase. Okay, so just earlier what we thought, when I was a student also like you, we thought always that these degradative enzymes are not important. So we should always work it on the inducible enzyme, synthesis enzyme. So they are more useful. But I tell you, most of the papers in the science or nature are from the degradation part, right? So mind it, because I'm using this word by my own experience. So cytokinin oxidase, first paper by Akishia and their group in 2005, they published a paper through CKX just by estimating the amount of CKX in a plant system, in the rice system, right? And they published that paper just to integrate with the help of IPT enzyme. So just the estimation of this degradation enzyme, we can say if less CKX is there, more production will be there. High CKX will be there, low production will be there, right? So that's such a small thing. So we should not use very high-fi science of research for these kind of studies. So that's the reason we started our research through cytokinin, and against that, cytokinin is the, this ethylene and jasmonate, where they are promoting the process of senescence, right? Yeah, this is the important slide I was talking about. So both are interacting with each other, the staggering trait as well as the stem reserve mobilization. So both, if more staggering trait, there is no study or no need to translocate whatever you have stored during your healthy days, right? If you are young enough. But for us, what we have accumulated during the younger time, that will be utilized for our future days, right? So similar kind of experiment, delaying senescence, increasing total photosynthesis, and can increase the graining. Perfect, fine. This is under the normal situation. But delaying senescence can also reduce mobilization. So no mobilization will take place if they are staggering, in nature. So there is a conflict of interest, right? But we have to use these two terminologies and try to find out the best possible way. I will come to that part. So this was just, yeah, this is one group, the Plumwall and his group. They published one paper, delaying senescence can enhance the drought tolerance. So this is wild-type well-water plant, senescence-induced cytokinin synthesis, right, under drought stress, and wild-type drought stress. So these kind of system they produced, and they showed the importance of cytokinin. So on that basis, we also utilize these kind of information. Senescence affect the post-harvest food quality also. If we can delay the post-harvest senescence or post-harvest ripening process, we can have the much more things in our hand for productivity or production point of view, right? And this is very old style, 2009 or 10 maybe, I think. So tackle senescence affects $100 billion industry. So you know, Netherlands is the capital of flowers. So that particular country and some of the other important European countries, they are making more than this now, more than this. So that's the importance. And this is a slide from the CIMIC, the Matthews Renault. I think that those who are working in the case of wheat crop or the cereal crops, they must have heard the name of Matthew Renault. So he's a physiological breeder, basically. So, and we utilize reserve mobilization and stay green for our future work. And first of all, that we started one of my master's student in 2014. We started from 14 to 22 till date. So I'll tell you the story from this time. So we also hypothesized delaying leaf senescence will enhance the drought tolerance. So we started with drought, converted into heat, and then combined stress. That is also important because plant cannot differentiate between drought and heat. So they will come across these two kind of stress at one time, right? So how we can spare these kind of systems? So we have to analyze, or we have to make some kind of system or create some kind of system where we can study both drought and heat simultaneously. So that's the reason we started this one. I'm skipping these kind of slide because of cheers. So physiological characterization of stray green trait in wheat germplasm under moisture deficit stress. So one of my first PhD students started working on this, and they showed by utilizing the leaf senescence rate in 70 wheat genotype, but we were not very happy, frankly speaking. Reason being, only 70 genotypes we could be able to take. So this was a field experiment, and he did some of the the drought susceptibility index. We categorized accordingly, and we categorized all these into the 4 boxes you can see, and we isolated the IPT gene from the wheat system. So after that, we started working on the characterization of these stray green traits completely in the wheat plant system. So not going to detail of this, but here also the IPT as well as CKX both plays a major important role, and whole signal transduction pathway we deciphered, and we presented the results. I understand it will be difficult to understand within this 20 minutes of time. So, but this particular slide I would like to show you that cytokinin plays an important and complex role in inhibiting stress response. Why I'm telling you, under moderate stress, it is showing higher expression, cytokinin. Under severe stress, it is showing very low cytokinin level. So this was really interesting story. We got it, we proved it by the TZ content estimation and certain other kind of estimations. We did it. We did this kind of experimentation, biochemical observation, not going to detail of each and everything. But this endogenous cytokinin level under the slow ripener or the slow senescence genotypes as well as the higher senescence genotypes, these contrasting genotypes are showing these kind of results. So we were very happy. Now we are coming into the details of it, and then further we dissect these two kind of contrasting genotypes for our further studies. The photosynthesis and related parameters, these are supposed to be concluded because we have to publish these kind of papers and we have to show various kind of evidences. So we did these kind of And this is the we decipher the cytokine signal components also. Not going to detail, but as expected, we got the results. So I can easily say like that also. So this is the final schematic depiction of cytokine-induced gray-green traits and its association with the drought tolerance in the wheat. So he contributed in that fashion, and we published this paper in field crop research. So being from the Agricultural Institute, we are supposed to work for 2 years, and that student got the field crop research paper. So means basically, and then the another PhD student we got were the ethylene and polyamines. So ethylene and polyamines, there is a, you know, the one quote is there, very important, the tug of war. So between these two, ethylene and polyamines, there is a tug of war for the basal substrate level. So they are fighting with each other with the help of SANDBC, sand decarboxylase. So that particular enzyme is important, and I'm not going to detail how much time is there. Okay, thank you. So because I would like to come to, so another PhD student, they make a formulation of this ascorbic acid, cytokinin, and paclobutrazole. So because in one of the experiment we have did this kind of individual experiment, then he formulate all this the experimental doses, and he created this, and he did these kind of evidential studies, and so next we published that paper in photosynthetic research, photosynthetica, and Cynthia Hall. So in that 3 papers he published out of that work. But the major point, now we wanted to convert our thinking towards the stem reserve mobilization. So again, now this stem reserve mobilization and staging, now I'm using these 2 words again, right? These 2 terminology for better flow rate fertility in wheat under combined heat and drought. So again, now from I have skipped the heat experiment, only drought experiment I have shown you. So these are some of the combined heat and droughts that we wanted to do it. And the novelty of this particular project is answer that staging trait and SM are negatively associated. That's what I was pumping on this particular point, right? Negatively associated, or it is possible to break the negative linkage. Now we want to break this negative linkage. That's what I told you, right? You understand that particular point? So now we want to break this linkage through pyramiding these traits for enhancing resource availability program. So how we can break that? That can be possible. That's what I thought about, and we got it finally. That is possible through the recombination or the mapping population. Through the recombination or mapping population approach. We did that, and we got the, this is 2 of the contrasting genotypes of the stragreen as well as SRM mobilization experiment from our previous study, and we crossed these 2, and we got the various kind of real population, recombinant in red lines, right? So 2 minutes, right? So we got this kind of information. So from there onwards, we started working with the 4 kind of conditions. We started working rain-fed, there is no irrigation, restricted irrigation, normal zone, and late zone. So these 4 conditions, we started working with this, and we started with the CIMIT panel association mapping with the help of 298 real population. So our genetics and breeder friends collaborate with us, CIMIT people collaborate with us, and we came into conclusion of these kind of experimentation. So not going to detail of this. Okay. So from the last year onwards, we started a work on stem reserve mobilization, stray-green trait for yield stability, again under heat and drought stress. Again here, the 280 real population we got in, and we started working on this. And finally, these 4 conditions we worked as earlier, and this is the most important slide which I wanted that you should concentrate. So under drought condition, we got the most stem reserve mobilization movement. This is the first one. After that is the heat one. The heat and drought one. And the third one is the control one, and the last one is the heat one. So stem reserve mobilization from the heat is very, very low. So because I met one of the pioneer of these kind of stun reserve mobilization work in the form of Blum. He's, right now he's 91-year-old from Israel. So I met him in Germany, and he told these kind of, without doing any experimentation, he told me these kind of results you will get out of this. So wonderful, heads off to that particular fellow. So that's where the experience now. So because they did smaller kind of experiment in their pot culture system, and they showed these kind of work. Not going to detail of what we got. So thank you very much. So this is my team, which produced a wonderful result. I'm not going to detail of it. So thank you very much for your patience hearing, and I'm ready because in 1 or 2 minutes maybe we are having. If you have any questions, really happy to interact.
And you put your sign, so you will be around lunchtime. Please ask.
Nobody will come, sir. I'm sure.
Thank you very much.
Thank you, sir. Thank you.
Thank you so much, sir, to pave a way to staying alive or green in the journey of sustainable agriculture. I would request the chairperson to facilitate our invited speaker, Dr. Ajay.
Thank you.
Our next invited speaker is Dr. Kishore Gaikwad, is an eminent principal scientist at NIPP, New Delhi. He done his PhD in molecular biology and biotechnology and postdoc from USA and Japan. His research laboratory is working on structural and functional genomics and transcriptomics of mandated crops. He has 90+ research articles in many reputed journals.
Shall I start? So, respected Professor Chakrabarti, Professor M. K. Reddy, and my colleagues on the panel, and the audience consisting of mostly young students and scholars. So, I'll be, after listening to the previous 3 speakers on carbon sequestration and stay-green phenomena, and a huge amount of work that we saw in the first presentation on genome editing. I'll be slightly diverting into genomics of a very little known crop by the name of cluster bean. उसको हिंदी में हम लोग गुआर कहते हैं. Not with that meaning which we refer to that word with, but it's a very small crop grown in a very small area in this country. Why I said it's a huge foreign potential, because it is one of the top 10 commodities which we export out of this country in the agriculture sector. So I started this work around 6 years back on deciphering the complete genome, transcriptome, whatever governs the basic traits which give us a lot of money in terms of foreign exchange. And that trait, we have been able to decipher the whole pathway. And now we are able to manipulate and regulate the various mechanisms. So this crop, cluster bean or guar, it's an Indian system crop. It originated in India and now in Pakistan also, some areas. 80% of the crop is grown in Rajasthan and Gujarat, and also consumed mostly in these states. It doesn't need water. 1 or 2 irrigation is enough. It's a very small genome. It's a very small crop. It matures in around 90 days or 100 days. And it doesn't require any chemicals, any fertilizers, any pesticide. It's a very hardy crop. So we picked up this crop for the reasons that I'm going to depict in the next few slides, because it earns us close to $1 billion every year. There's a huge potential in the international market. And at a biochemical level, this crop produces what we call as guar gum or galactomannan, which is a natural hydrocolloid. Like it has immense potential in drug delivery and those applications across the pharma sector. So the guar gum is nothing but it's an endosperm which provides a lot of nutrition to the growing embryo. It is taken from the seed of the guar crop. And apart from pharmaceutical, it has immense potential in various industries, including cosmetics as well as food industry. And now it's also being used in shale gas mining in Europe and the US. So this is the basic structure of a guar gum. This is basically a galactose mannose backbone. And this random distribution of the galactose molecule on the mannose polysaccharide leads to creation of a guar gum molecule. Now, gum is produced by many crops in many plants like carob and locust bean and even fenugreek. But the gum produced by this particular crop is of the highest nature in terms of quality as well as its physiological properties. So we picked up this crop for complete analysis around 6 years back, and this is my kind of first exposure. So we are kind of announcing this crop in its complete genomic assessment. So this guar gum, one of the major properties of a guar gum is that it is highly soluble in water. Even at freezing temperatures, around 3 degrees or 4 degrees, this will dissolve rapidly in cold waters. It's biodegradable, of course, and it resists all kind of chemical and all those oil treatments. So we started decoding the genome in 2016. We had a funding from ICR, and this is an orphan crop, sustainable agriculture, sustainable development, human welfare. Everything is being packed into this crop and its cultivation practices. So we started investigating when nothing was known about this crop. So I'll be telling the whole story in these 5-6 points in the next 8 to 10 minutes. So we started in the unknown area, starting with the genome size. We did a lot of flow cytometry. That's a basic thing we should do before we take up any genome. And it comes around 550 million bases. So it's slightly bigger than rice and lesser than the other legumes that I work on, like pigeon pea. And then we started investigating into the plastids and the mitochondria and analyze the whole genome in that totality, because these are important contributors from a physiological perspective, from a sugar perspective. And then I'll just slide through. Then we picked up close to 150 to 200 accessions from Rajasthan and Gujarat. We have a sister institute in Jodhpur who helps us with all the breeding part. And then we realized that the diversity that this crop harbors in this country, in the cultivated gene pool, is very, very low. We need to scout for more of the guar wild accessions across the country. And we don't see many people working in that area, but we need to tap the resource since now we have this particular genome in our hands. We did then also we tried to understand what is why the gum is accumulating in the seeds of this particular crop in conditions which are very, very high temperature and low moisture conditions like Rajasthan. There are differences between a set of genotypes. If we see here in the 2 genotypes, there are differences on the cuticles and the palisade cells. So this is where the gum content keeps on increasing when the seeds are growing or the seeds are developing. And the varieties which have a low gum content, they are mostly filled with air on the left side, and the ones which are high gum content, they are tightly packed in the vacuoles, because this is where the galactomannans are stored. Then we started creating a transcriptome, as the earlier speaker also mentioned, that we need to create a resource of genes and all the variants that come into the picture when we pick up a particular trait. So we kept on accumulating data from all angles in this particular crop, and then we picked up 2 genotypes These two are differentiating in their gum content, in their growth parameters, in their cultivation habits. And the main issue is the gum content. The gum content in one of the varieties is 14%, the next is 32%. So we always believe in mapping and breeding and molecular analysis. So we picked up these two genotypes, made a cross between these two genotypes, made a mapping population, and then we could map the entire pathway of the galactomannan biosynthesis in these mapping populations. Then we did investigations. There are around 40 genes in these galactomannan pathways. We picked up all 40 of them. We did analysis across all the 150 genotypes that we had. We mapped them. We did a structural analysis. We went into their functionalities. We went into their SNPs and indel structures. And we then came down to a set of transcription factors which control the regulation of this pathway in cluster B. And this is a universal pathway which we have seen to occur also in other species which create such kind of chemicals. So we had this, we picked up, we have 3 compartments now where we know that this is a stage where the gum accumulates, the galactomannans are being produced inside the developing cell within the seedling, in the developing seeds. And these transcription factors now we have already picked them up, and we are trying to overexpress and see how they make the gum content better. To increase it to even by 10 or 20% will result in a huge potential in terms of foreign exchange. This is the whole pathway that I'm speaking of. We could analyze the whole galactomannan pathway. It's a very complicated pathway. I can't describe too much in detail here, but this pathway has the potential to produce more gum in different genotypes, in different conditions, and that will result in a better and a more sustainable development for the farmers and also for the gum industry. Then we also analyzed the small RNAs, and fortunately we could identify some microRNAs which were targeting the 2 genes, the mannan synthase, which is a main gene which controls the entire gum synthesis of the galactomannan pathway. And we are in the process of now taking these further through the editing techniques or overexpression. The only issue is this crop is top 10 commodity crop. It's imported, it's exported outside. We will not touch it very easily until we get all the safety and regulatory permissions. Then we started, we had already started the genome. The genome went on well. We employed all the chemistries that we had. But since we did the rice genome, we always believe in map-based sequencing. So we created a linkage map first, a so-called SNP map between 2 genotypes, again at our Jodhpur center. And then we did the sequencing and aligned all the data on the particular map. And then we could assemble the genome into 7 chromosomes. This is one of the best genomes, especially in the legume section, better than chickpea and pigeon pea and other genomes that are closely related to these particular species. And this particular genome, very shortly we're going to announce this genome, maybe in a couple of weeks or months time. We did a molecular clock analysis, evolutionary perspective to find out what is happening or how this particular crop originated, and did all the ancestor analysis and so. And then we analyzed that it has lost around 9,000 genes as compared to soybean and pigeon pea and other legumes, which are called as a master genomes or ancestral genome. And then now we are at a stage where we have all the data with us, and we want to go forward and use this data to create an environment where we can increase the foreign exchange capability, and help the farmers in the society on a larger basis. So this guar gum has a, if you any of you go to the Bombay Stock Exchange or the National Stock Exchange or Nifty, there is a guar index. There's a guar gum index, if you are very savvy with the investments, this is the area where you can play some bets. And in coming times, this is going to be a big commodity as far as listing and sharing of the trades is concerned. This is an estimate by different agencies which say that the guar market is estimated to grow by $1 billion or more every couple of years. And this is where, as a country, as India, we should take the lead, especially since we have all the data and the information with us at this stage. I'll wrap it up, sir, here now. Thank you.
Thank you so much, sir, to make a base of genomic insights in the building blocks of sustainable agriculture. Now I would request our chairpersons to please facilitate Dr. Kishore Gatwar.
Yeah, we have the number. It's already published, sir. It's already there.
Next invited speaker is Professor A.B. Nadar from Savitri Phule University, Maharashtra, whose research work area covered plant tissue culture, plant molecular biology, biochemistry, and molecular genetics of basmati rice aroma. He has 70+ research papers and 5 books, and has received a number of honors and awards.
Good afternoon, respected chair, distinguished scientists and student friends. It's my pleasure to share the work that is going on in my lab. So my lab basically is working on basmati rice aroma volatiles, and then pertaining to the theme of this conference, I thought of sharing the work that is we are doing with respect to the sustainable approach to enhance the rice aroma. So all of us know that rice is the cornerstone of food and culture, and then the first food grain that the kid eats is the rice grain. Okay, so that's how it is very much well associated with our dietary habit. So for centuries it has been remained as a cornerstone of their food and culture. So coming to the basmati rice, is a natural gift to India. We know that basmati rice means the queen of fragrance rice, queen of fragrance, or the perfumed one. So all of us know that it is a long-grained rice with fine texture, and it is one of the costliest rice in the world. And the another beauty of the basmati rice is that lengthwise it elongates more than the breadwise elongation. And this is a very peculiar trait that is associated with this basmati rice. And then the added feature is that this has got a pleasant aroma. In addition to basmati rice, we are having the upland rice varieties, and these are categorized as the non-basmati type rice varieties. So yesterday's lecture by Professor Khanna, he was referring to some of the rice varieties, especially Kala Namak. So these are the rice varieties that are not falling under the category, but they are falling under the non-basmati category, and then they are short grain type or medium grain type, and then they are having brilliant taste. So they don't share the properties or characteristics of basmati rice, but in terms of cooking, but they are having the pleasant and very brilliant aroma. Sometimes it is more than basmati rice also. And then another important feature is that the common man can afford this rice. So in compared to basmati rice, these non-basmati rice varieties, they are affordable by the common people. In addition to that, they are enriched with high protein, contains fibers, and we are having the wide range of rice varieties, these non-basmati rice varieties that are being cultivated by our local farmers. So, as all of us are knowing, India is the major producer of rice. So, the major share, 85% is comes from India, followed by Pakistan, and these are the countries that are involved in importing the Indian basmati rice, so mostly the Middle East countries and other of the European countries. So, these are the figures of basmati and non-basmati rice in terms of export. So you are seeing here that in comparison to basmati, non-basmati rice is export in more quantity. So these are the values that that India earns through the basmati rice export. So $3.53 billion of amount is earned by the Indian government. So in addition to that, these are the recent figures that we are having with respect to the non-basmati rice export, and then almost $6,115 million of the foreign currencies earned through this through the export of this non-basmati rice. So if you look at the statistics, the data tells us that there is a jump in the export of non-basmati rice from 2013-14 to the last year. So this is by 109%. So this indicates that there is a high demand for these non-basmati rice. So coming to the scientific aspects of this, that very interestingly, flavor or aroma, it is constituted by more than 100 volatile compounds, and then you are seeing here these are the chemical classes that are sharing in the pleasant rice aroma. So the aldehydes, alcohols, acids, hydrocarbons, phenols, esters, and ketones, so all they contribute. And what perception we get as a rice aroma is the total combination of these chemical classes and compounds. So very interestingly, among these, the inventors, they have come up with very unique compound, and this is called as a 2-acetyl-1-pyramide, abbreviated as 2-AP. So we are seeing here, this is the chemical structure of 2-AP having methyl ketone group and pyrrole ring. So this compound has been identified, 2-acetyl-1-pyrrole has been identified as a unique aroma compound, and then irrespective of whether it is basmati or non-basmati rice, it is uniformly present in all the aromatic rice varieties. In other words, this is the compound that gives the aroma, typical aroma to the basmati rice and fragrant rice. So, it has got a pleasant popcorn-like odor. The odor threshold is very low, 0.1 parts per 10⁹ parts of water. And then another interesting thing that this particular compound is a highly unstable compound. The pyrrole ring makes this compound very unstable, and that's why in the market we do not have essence like basmati essence. Okay, like how we are having vanilla essence or other essence that are available. The synthetic essence or synthetic compound is not available because of the very low shelf life. So this is the biosynthetic pathway of S1-pyrrole, and that one lab from Australia, they have worked out that there is one gene that is a beta-naldehyde dehydrogenase 2 gene, and this is involved. This gene gets mutated. There is X base and 3 SMP deletion. This gene is responsible for the accumulation of 2-acetyl-1-pyrrole. So one of my students is talking on that, so she will explain this in detail. So very interestingly, can we ask this question that whether this particular 2-acetyl-1-pyrrole is synthesized in basmati rice only, or whether the rice aroma is present in only rice? You know that nature is the wonderful creator, and then you will see that in addition to rice, this particular compound is synthesized across wide range of organisms. So very interestingly, you are saying that this is the tiger, and then in the urine of tiger, this particular compound is synthesized as a pheromone. Now, the another example that in the lower organisms also like bacteria, this particular compound is synthesized with Bacillus cereus. Then, in addition to that, in fungi also, how the fungal species like Aspergillus or mori, this particular compound is synthesized. So this, you must have smelled the pleasant flavor of the popcorns. So, through the chemical reaction, what we are calling as a Maillard reaction, this particular compound gets synthesized. And then, there is a variety of mung bean called as Sona mung bean, cultivated in the North Bengal. And this particular mung bean also, synthesizes this particular 2AP. And then in the Basia latia flowers, what we are calling as Maua flowers, also this compound is present. Then Vallis glabra bread flower, then Pandanus amarylifolius. So all these are the other plants and species where you will find this particular basmati rice aroma compound 2AP is synthesized. So on this particular plant, Pandanus amarylifolius, my lab has worked extensively. So this is in nutshell that how this basmati aroma gets synthesized all over various food products. So all of us are interested in having this basmati rice, okay, and it is widely used and popularly used. But if you go to market and if you buy a good brand, good quality of basmati rice, and then if you cook it to make some of the foodstuffs like biryani and other things, no doubt that the other properties of basmati rice will be there, but in terms of aroma, you might experience that the aroma is not there as much you were expecting. So suppose you are paying more than 200 rupees a kg for a rice, basmati rice, and then the aroma is pleasant aroma is not there, or you are missing that aroma. Okay, so what has happened to that aroma? So some of the things that we worked out, that the use of inorganic fertilizers, the change in the agriculture trend from traditional to modern, and then the use of the pesticides. These are some of the factors that were identified as the reason for depleting the rice aroma. So obviously, we thought of looking at these aspects of how we can improve this aroma. So, in my lab, we have defined some of the strategies that can enhance the rice aroma. Some of the biotechnological approaches, transgenic approaches that we practiced or we worked out in my lab are pathway engineering. So, where we overexpressed the one of the gene P5CH gene, which is responsible for synthesizing proline, which is the precursor for this particular 2AP. So we overexpressed that gene, and then we got enhancement in the rice aroma. Second approach was in non-scented rice through gene silencing approach, mRNA gene silencing approach. If we will silence the RNA, then that gene gets silenced, and then we can have the expression of aromatic. So we did this in IR64 successfully, and then we are having the transgenic IR64 line with 2AP or that aromatic line. And then the another one, that is non-transgenic approach, which is the focus of my today's lecture, is that is the rhizosphere enrichment. So very interestingly, what we did that we worked on the rhizosphere of these basmati rice, and then we isolated some of the bacterial strains that are synthesizing the rice aroma. So we all know that rhizosphere is a dynamic zone surrounding plant roots and has a great significant impact on ability of plant to capture both water and nutrients.
Now we have oral presentations from young researcher. Firstly, I would request Sadia Ikrar from Jamia Hamdard. There is time limit of 15 minutes.
The audience, please ask questions because these three lectures are there. These are competitive lectures. So you will
That would be
see
references.
there
Thank you.
and appreciate the network. Please ask questions.
Good afternoon. Myself, Sadia Ikrar, pursuing PhD under the supervision of Professor MZ Abdeen. Am I audible? At the back? I'm pursuing PhD under the supervision of Professor MZ Abdeen, Department of Biotechnology, Jamia Hamdard. I'm presenting here on microRNA potential regulator of genes involved under sulfur uptake and assimilation pathway in Brassica gentiana under sulfur treatment. Brassica gentiana is an important winter season crop grown in an arid region. It is the second most edible oilseed crop after soybean. They are mainly grown for leaves and seeds, which constitute an important source of fats and various vitamins. Cultivars are mainly grown for its oilseed content, which contain palmitic, pantoic acid, and various other fatty acids, and it demands high amount of sulfur when compared to the other crops. What is the need of sulfur in this plant or any other plant? Sulfur is necessary for synthesis of amino acid, proteins, and various cellular components, and the secondary metabolites, including the sulfur compounds. Sulfur is part of amino acid cysteine and methionine, which are the building block of various protein necessary play a crucial role in activation of various biochemically active enzymes. Overall, the diversity and the amount of sulfur can influence the quality of crop and also affect the nutritional value for human and animals. However, the deficiency of sulfur can alter the oil content and also affect the efficient use of other nutrients, especially nitrogen. There is a metabolic coupling between nitrogen and sulfur at the level of O-acetyl L-serine. Now, the factor affecting the growth of Indian mustard could be exogenous and endogenous. Exogenous are sulfur, nitrogen, and heavy metals, and endogenous are microRNA, endogenous siRNA, and various other non-coding RNAs. Here we focus on the microRNA. They are the class of short endogenous non-coding RNAs. They base pair with their target to repress their translation or induce degradation, and in certain cases, microRNA have also been reported to enhance the expression of their target genes. The pathway of sulfur assimilation began with the uptake of inorganic sulfate, which translocated to the chloroplast, where it assimilated into APS, then sulfate, sulfide, cysteine, glutathione, and also with methionine. And at the excess of sulfur, mobilized to the vacuole, and at the time of need, it remobilized back from there. The uptake process is driven by the sulfur transporter. They are high affinity sulfur transporter and low affinity sulfur transporter. Now, the proposed hypothesis, the rise in global population, new emerging economies, and exponential increase in food demand implies an urgent need to increase the agriculture production. But the use of fertilizer to enhance the crop is not a remedy, because the optimum nutrient level above the optimum level can affect the plant growth and also contaminate the environment. Therefore, there should be other ways to enhance the sulfur assimilation in Indian mustard. Until date, no experimental validated research activity have been reported for exploring the microRNA involved in the sulfur uptake and assimilation pathway in Brassica gentiana. And this will limit the use of chemical fertilizer also and improve the entire quality of the crop plant if we target its uptake and assimilation pathway. Now, the workflow. Firstly, we have obtained the genes involved under the pathway from NCBI. They were uploaded in psRNA target analyzer and the user submitted transcript. 39 targeted microRNAs were identified. and some of the microRNA from attained from the mill base. They were again submitted in Miranda database tool, and we have predicted the 10 highly conserved microRNA validated using the stem loop RT-PCR. Table showing the data obtained from the Miranda database tool, and these 10 microRNA were selected on the basis of their minimum fold energy and percent complementarity, also showing the target genes and the inhibition, whether it's translational or cleavage. Then the plants were grown in a sulfur treatment, insufficient, deficient, and high condition. And the total RNA was isolated from the 3 cultivars, and the 3 stages were selected: vegetative flowering and pre-flowering. The primers were designed which are specific to miRNA and mRNA cDNN. Then the quantitative real-time PCR was performed for the validation of expression of microRNA and the target genes. We have also validated the target of mRNA. They are authentic targets of the microRNA through a 5' RLM base. And the metabolite and sulfur content was also estimated through GCMS and CHNS analyzer of the treated sample. The result showing the real-time analysis of selected microRNA under the treatment in 3 stages in PUSA bold. Here, the 10th microRNA, which is microRNA 159A, showing up regulation in both the treatments. MicroRNA 395 in sulfur insufficient condition, and second microRNA 865 was upregulated in both the treatments. Similar kind of expression pattern was obtained in PUSA, Jakisan, and Varuna, but there is a fold variation. Now, the fold change in expression of genes and sulfur transporter quantified by real-time PCR in all the 3 cultivars under treatment in leaf and root tissue. Here, the ATPase gene was upregulated in sulfur high condition, and the sulfur transporter 2,2, which is a low affinity sulfur transporter, is upregulated in sulfur insufficient condition in leaf tissue and downregulated in root tissue. Then the 2 sulfur transporter 4,1 and 4,2, which are chloroplast transporter, are upregulated in sulfur insufficient condition. In Varuna, similar kind of pattern was obtained for the chloroplast transporter, and the sulfur transporter 1,1, which is high affinity sulfur transporter, and 1,2, which is also high affinity sulfur transporter, was upregulated in sulfur high treatment. In PUSA Jakisan, the sulfur transporter 2,2 in leaf tissue, which is a low affinity sulfur transporter, is upregulated in sulfur insufficient condition, and sulfur transporter 2,1 in root tissue also upregulated. Then we have done the GCMS. The heat map was generated through meta-analysis software, and we have focused on the free amino acid. Here, the cysteine in PUSA Bold and proline, free amino acid, were high in highest treatment. In PUSA Jakisan, the cysteine level was high in highest treatment, whereas the alanine and serine level was higher in insufficient condition. In Varuna, the free amino acid cysteine, valine, serine, isoleucine, and alanine concentration were higher in as insufficient condition than sulfur high treatment. Then the CHNS analyzer was used to estimate the sulfur content. The sulfur in post-flowering stage in PUSA Bold was highest compared to the other crops. The Varuna has high sulfur content at the sufficient stage, and PUSA Jakisan and PUSA Bold has sulfur high content in sulfur high condition. Then the coefficient relation. Then the correlation between microRNA and their target mRNAs was interpreted through Pearson coefficient. Here, the microRNA 395 is positively correlated with sulfur transporter 2,1, and microRNA 865 was positively correlated with sulfur transporter 4,1 and 4,2. Then also the target validation, the cleave side of target microRNAs, target mRNAs for conserved microRNAs were experimentally mapped by RLM-based assay. Here, the sample were treated with different enzymes, and the adapter was added, and the primer was synthesized for known as auto primer, which are gene specific and adapter specific. Then they were amplified, and the amplified product was again used as a template. Then the inner primer was synthesized for that template for the further confirmation. The gel electrophorese showing the cleaved amplified product, and it reveals the authentic target of microRNA. Then we have, on the basis of previous study, we have selected the 2 microRNA, miRNA 395 and miRNA 865. miRNA 395 targeting the multiple genes, including the key gene, and 865 is targeting the chloroplast transporter and the regular transporter. For the functional validation, we have used STTM approach, short tandem target mimicry. In this case, it has two binding sites to the microRNA and a spacer region at the cleave site. It will inhibit the expression of microRNA, and we have purchased these STTM vectors from the AD gene. They were provided it as a step culture. The culture was revived, mobilized to the agrobacterium, and they were validated on a yam agar plate containing the antibiotic kanamycin. Then the single colony was pegged, grown in a liquid medium. Plasmid was isolated and validated through restriction digestion. The lane 1 and 2 has a control. Lane 1 and 3 has a control vector of STTM 395 and 865. And lane 2 and 4 has digested vector with BST2 and BAMH1. Then the agrobacterium containing the vector of interest will mobilize transiently transforms in leaves of the 3 cultivars. And the RNA was isolated at 2, 4, 6, 8, and 10 days of post-infiltration. Then the heat map was, cDNA was synthesized for microRNA and mRNA, and the real-time analysis was performed for their expression studies. We have got the best result at the 6 days of post-infiltration, where the expression of the genes is upregulated if it's positively correlated with the microRNA, and if it's negatively correlated with the microRNA, and the expression of microRNA is downregulated in both microRNA 395 and microRNA 865, like STTM 395 and STTM 865 case. From these data, we have interpreted that the overexpression of ATPase when we downregulated its targeted microRNA can enhance the sulfur assimilation or sulfur incorporation from the soil. And even the downregulation of sulfur chloroplast transporter 4.1 and 4.2, when we have downregulated its positively correlated microRNA, can also, through feedback mechanism, can enhance the uptake of sulfur from the soil. So that for the future work, we have planned to develop the stable lines. For that, we have used a floral dip transformation approach, and the bud was selected. So the entire process, the seeds has been collected, and we are working on that. Now to summarize my work, microRNA involved in the regulation of sulfur uptake and assimilation pathway in Brassica ginsea were identified using PSRNA target analyzer and Miranda database tool. They were predicted 10 highly conserved families, and using stem loop PCR, they were validated. Then the 3 varieties were grown in a sulfur treatment, and their expression analysis was studied. The correlation between them was also studied, and the influence of sulfur on metabolite and sulfur content was also estimated using GCMS and CH analyzer software. The 2 microRNA selected were functionally validated through STTM approach, and our study showed the potential role of microRNA in the regulation of sulfur uptake and assimilation pathway, putting forward the implication of these molecules to enhance sulfur content in the Brassica ginsea. These are the references I have cited, my publications till date, and I would like to thank my supervisor for his support and guidance, and I also thank my colleagues. Thank you so much.
Queries, please.
Absolutely, and even we have 2 minutes time and couple of questions. Appreciate you that.
Do you have any data on bacterium transformation as well as floral dip transformation?
Sir, I have data with me with that. I have just after floral dip transformation, just the seed has been developed. Now it's a sowing season of Brassica ginsea. We have sown the seeds.
Means the regeneration efficiency differences are there.
Regeneration efficiency of dealing with that. Can you elaborate more?
No, means the efficiency. Why not start with the agrobacterium transformation? Why you use transformation?
So just to develop the stable line, I have meanwhile developed that another method. Yes, sir. That floral dip method also contains the acidocyanidron and agrobacterium. It also has that, and the other process we have also doing that simultaneously. But my favorite method is floral dip because it's already developed in our lab. Pardon. My for sulfur sufficient is 1 millimolar, already known in the literature, and for sulfur high is 4 millimolar, and for sulfur insufficient is 50 micromolar. For sulfur sufficient is 1 millimolar. No, I didn't done any physiological. It's already known in the literature that this is the desired concentration for all the treatments. My focus was on molecular studies, just to target the microRNA.
So my name is Dr. Jana. I'm from MSc Biotechnology first year. So my question is, how is the Pearson correlation is
Interpreted. Okay, it was studied. Okay, it was interpreted through meta-analysis software. For that, the values obtained, the expression values obtained from the data-later CT method, which I have done for the RT-PCR, was submitted in that software, and it provided the values. Right.
Why are you choosing 3 varieties only?
The 3 varieties are depend on their sulfur utilization efficiency, which means like the limited amount of sulfur the plant needed. The 3 varieties, among them the sulfur Pusa Bold is highly sufficient, and Pusa Jekisan is moderately, and Pusa Jekisan is insufficient, and Pusa Varuna is moderately sufficient. It depends on how much minimum quantity they can grow.
Next oral presentation is from Sonali Chandanshi from Savitri Phule Pune University, Maharashtra. Hello, good afternoon everyone. Myself Sonali Chandanshi, and I am doing PhD under the guidance of Professor Abhinada in Savitribai Phule Pune University, Maharashtra. Today I am presenting my research work, that is DST mutational analysis, a key to predict genotype to phenotype association and tailoring rice varieties to accelerate breeding approaches for sustainable agriculture. So many speakers has talked about rice and its nutrition quality. So rice is a major staple food for most of the world's population, and it has high nutrition quality. And rice is agriculturally important commodity with third highest worldwide production after sugarcane and maize. So Indica japonica and javanica are the subspecies of rice, Raja sativa. Among rice, aromatic rice is nature's gift to India because of its aroma. Aromatic rice includes basmati and non-basmati. Basmati has long and slender grain. Grain elongates almost twice upon cooking, has a high price. Some example of basmati rice, that is basmati 370 and Pusa basmati. Non-basmati has medium to short grain. It is available at reasonable price, and non-basmati rice has high diversity. Some examples are Ambe Mohar and Kalanama. So question comes that what contributes to the aroma? More than 200 volatile compounds have been identified from the aromatic rice grain, which contributes to the aroma. Amongst these volatile compounds, 2-acetyl-1-pyrrolene has been identified as a principal aroma compound. This is the structure of 2-acetyl-1-pyrrolene. This is the 2-AP biosynthesis pathway, in which proline, glutamate, ornithine are precursors of 2-AP, which will be converted into P5CS, which is, this conversion is taken by P5CS. This P5C is then converted into delta-1-pyrrolene, which is in equilibrium with GABA. When BADH2 is functional, GABA is converted into GABA, and when BADH2 is non-functional, GABA
delta-1 pyrroline reacts with methyl glycol to form 2-AP. And expression of 2-AP is linked with down regulation of BA-2. Means in aromatic rice, BA-2 is non-functional. So there are many reports. One of these is 8 base pair deletion in exon 7. So functional markers are markers which we use to amplify genic region. It enables efficient and quick characterization, and it is used to screen allelic diversity. So two objectives are framed for this work. That's screening indica rice accession to explore allelic diversity in using two functional markers, that is BA-2P 5' UTR and FM-BA-2 exon 7, which amplifies BA-2P 5' UTR amplifies the sequence in promoter region, and FM-BA-2 amplifies the sequence in exon 7. And to study the correlation between BA-2 mutation and 2-AP biosynthesis. So these are the rice varieties I have used in these studies, which includes scented non-basmati and basmati. And I have selected 3 non-scented rice varieties to compare the sequences, which were collected from Maharashtra, Karnataka, and New Delhi. For this screening of rice accession with functional marker, for which I have isolated DNA from the selected rice accession, then PCR amplification using 2 selected markers, then gel electrophoresis, then sequencing of selected rice accession, then sequence analysis. This is the representative gel pictures, where first picture is showing DNA on agarose gel, second PCR amplification with BA-2P 5' UTR, and third one is PCR with FM-BA-2 exon 7. By scoring the gel, we have differentiated this rice accession in 4 categories. First, in which mutation is there in 5' UTR also and in exon 7 also. Second category is mutation in only 5' UTR. Third one is mutation in only exon 7. And fourth is there is no mutation in either of the region, that is 5' UTR or nor in exon 7. And then sequence were analyzed. In 5' UTR, we have observed 250 base pair deletion, and in exon 7, 8 base pair deletion and 6 SNPs. And in Lal Bhat non-basmati rice variety, so we have observed only 1 base pair deletion in exon 7. So these are the mutation revealed in aromatic rice in this present study, that is 250 base pair deletion in 5' UTR, 8 base pair deletion and 6 SNPs in exon 7, and 1 base pair deletion in exon 7. So after this correlation between BA-2 mutation and 2-AP biosynthesis, for this I have selected proline, methyl glycol and GABA metabolites, and these metabolites were analyzed. Proline were analyzed using Bates et al. Method, second methyl glycol Yadav et al. Method, and GABA is Arlide and Sudjog method. Then BA-2 and P5CS enzyme activity were carried out using Vapte et al. And Garcia-Reyes et al. Method. For the gene expression, we have isolated RNA, then cDNA synthesis, then QR-PCR analysis, and analysis using 2-delta-delta CT method. These are the results of metabolite analysis. Proline is high in scented rice variety Kagesali, Lal Bhat and Kala Krishna, and GABA is as compared to scented varieties. Proline methyl glycol is less in non-scented rice variety. These are the results of BA-2 and P5CS enzyme activity. But in scented rice varieties, P5CS
enzyme activity is high and BA-2 enzyme activity is low. And in non-scented rice, P5CS enzyme activity is low and BA-2 enzyme activity is high. So, this is the result of BA-2 and P5CS gene expression. Like enzyme assay, in scented rice, P5CS gene has high expression and BA-2 has low expression. And in non-scented rice, P5CS has low expression and BA-2 is highly expressed. So these are the conclusion. So using this functional marker, earlier report showed 8 base pair insertion in promoter region and 8 base pair deletion and 3 SNPs in Japonica rice. And in this study, we have observed 250 base pair deletion in indica rice in 5' UTR region and 8 base pair deletion and 6 SNPs in exon 7, as well as novel 1 base pair deletion in lal bhat non-fragrant rice varieties. And the present BA-2 mutational analysis, relative metabolism, enzyme and gene expression has confirmed the relation between mutation in BA-2 and 2AP biosynthesis. So this information gained through the present study can be utilized for marker-assisted breeding, genomic-assisted breeding, and the present mutation analysis extent is used to accelerate biotechnological approach. By this, we can convert non-basmati rice using biotechnological approach to aromatic rice, and we can improve aroma in aromatic rice through transgenic and non-transgenic approaches. Thank you. Queries, please.
Have you heard latest, very recently, first genomic
BA-2 mutation. That is BA-2.
through and well-conducted research, the Commission concluded that suppressing the use of herbal cannabis, bhang, would be totally unjustifiable. They concluded that its use is very ancient, has some religious sanction among Hindus, and is harmless in moderation. In fact, more harm was done by alcohol. Furthermore, prohibition would be difficult to enforce, encourage outcries by religious clerics, and possibly led to the use of more dangerous narcotics. These findings of the Indian Hemp Drugs Commission report of 1894, conducted over 100 years ago, are surprisingly relevant today. Despite lingering controversy, use of botanical cannabis for medicinal purposes represents the revival of a plant with historical significance re-emerging in present-day healthcare. Legislation governing use of medicinal cannabis continues to evolve rapidly, necessitating that pharmacists and other clinicals keep abreast of new or changing state regulations and institutional implications. Ultimately, as the medicinal cannabis landscape continues to evolve, hospitals, acute care facilities, clinics, and long-term care centers need to consider the implication, address logistics concerns, and explore the feasibility of permitting patient access to these treatments. Whether national policy will offer some clarity or further complication, regulation of this treatment remains to be seen. The most common use for medicinal cannabis include for severe or long-term pain, nausea, and vomiting due to chemotherapy, and painful muscle spasms. However, there are several medical conditions for which patients can use medical marijuana, which can vary by state law. Cancer-related pain, glaucoma, HIV, AIDS-related, weight loss or nausea, vomiting. Muscle spasms, neuropathy, Parkinson's disease, panic disorders, post-traumatic stress disorder, seizures, epilepsy, severe pain, chronic pain, not amenable to other treatments, etc., etc. As I stated, we are soon going to start cultivation of medicinal cannabis in the state of Uttarakhand at Pantgaon, district Almora. Besides this, we are also focusing into cultivation of industrial hemp. The hemp plant is a variety of cannabis sativa that has been cultivated for its fiber, not any psychoactive effects. Many strains of hemp have none of the mental effects associated with cannabis. This type of industrial hemp has been used as a fiber source, oil, and as an important source of nutrition for thousands of years. We are focusing on hemp fiber as we firmly believe that sustainability is the only way to build a livable and a lovable future for our generations. I would like to give a brief profile of hemp fiber, which is a far superior fiber to the commonly used cotton. Cotton is by far the most widely used fiber in the global garment industry. Around 60% of all cotton produced worldwide feeds into the fashion industry, approximately 30% for furnishing, and the rest for the industrial usage. More than 70% of the fiber consumed in the apparel industry are some type of cotton. Compare that with the hemp, whose myriad benefits are only just beginning to be rediscovered. There are still unexplored aspects to this miraculous plant. Hemp simply does not enjoy the same type of worldwide availability and usage as cotton. But we all know cotton is a highly water-intensive plant. From sowing to reaping and through the production process, a single t-shirt can require up to 2,700 liters of water. Cotton also relies on a barrage of pesticides and weedicides to grow. The overall yield takes at least 5 to 6 months to harvest. And 1 acre of land produces just about 500 pounds of cotton. Even if the farmer uses all natural fertilizers and pesticides to grow his crop, the conversion of fiber to fabric requires dozens of chemicals and synthetic dyes. Many such dyes are compounds of heavy metals and iron that take millions of years to degrade naturally. Cotton might be an alluring choice for adopting a sustainable lifestyle, but these hidden facts should caution you against the fiber. Hemp, on the other hand, is far cleaner and environmentally friendly crop. Consider this. Every single part of the plant, from leaves, stems, flowers, roots, etc., has some industrial application. Hemp finds itself used in garment manufacturing, health and nutrition, treatments, furniture productions, and construction activities. At the Hemp Foundation, we operate on a zero waste framework, that is, no part of the plant is thrown away. Hemp plants are natural habitats for weed-eating insects, so there is little need for supplying additional synthetic herbicides and weed killers. The plants are thin and tall, which means that they can grow more densely per acre than cotton. The plant's yield is phenomenal. A single acre can produce up to 1,500 kg of fiber. Hemp needs 40 times less water than cotton for growing, whereas 1 kilogram of cotton takes up to 20,000 liters of water in its life cycle. A kg of hemp only needs about 500 liters. All in all, as a crop, hemp is far more durable and environmentally sustainable than cotton. I'm entirely on board the hemp train thanks to its wonderful qualities that cotton simply cannot match. Hemp clothing is long-lasting, comfortable, breathable, and doesn't show age and discoloration. The best part is that it is a sustainable crop. When coupled with the proper manufacturing processes, it contributes to water conservation and removing harmful microfabrics from the water table and soil. To sum up, I wish to say sustainability is a societal goal that broadly aims for humans to safely coexist on Earth over a long time. Specific definitions of sustainability are difficult to agree on. The concept of sustainability can be used to guide decisions at the global, national, and individual level. Sustainability is commonly described must be addressed for a sustainability transition to become possible. Some sustainability barriers arise from nature and its complexity. Other barriers are extrinsic. To the concept of sustainability, a number of extrinsic sustainability barriers are related to the dominant institutional frameworks, where market mechanisms often fail to create public goods. Some example steps humanity can take to transition to environmental sustainability include maintaining nature's ecosystem services, reducing food waste, promoting dietary shifts towards plant-based foods, further reducing fertility rates, promoting new green technologies, and adopting renewable energy sources, while phasing out subsidies to energy production through fossil fuels. Global issues are difficult to tackle as they would require global solutions. But we all can contribute in whatever best possible way we can to make our Mother Earth a better place, to live for ourselves and our future generations. Thank you.
Thank you so much, ma'am, for a revolutionary and thoughtful talk and to pave a way with the help of hemp in the journey of environmental sustainability. I would like to request our chairpersons to please felicitate Dr. Geetika ma'am. Ma'am. Now in this session, we have 3 oral presentations. I would like to invite Mr. Rohan Chabra from Prakriti Agtech, Rohini, New Delhi for an oral presentation.
ये पॉइंटर है ये फोटो तो फॉरवर्ड ये स्क्रीन नहीं जो घूम के पढ़ाएंगे ना ये बैकग्राउंड फॉरवर्ड आएंगे
Sorry for this. So I'll start. So good afternoon to all. And first of all, I would like to thank the eminent personalities, the eminent panel members we have, and distinguished scientists we have today with us. And I would like to thank you all for joining us today, the young, bright minds with smiling faces. And so our topic is basically building climate smart agriculture infrastructure through innovation, and why there is a need of basically developing a climate smart infrastructure. What is climate resilient? What is climate change? Why, you must be wondering why I'm talking about climate change, right? So I must tell you that if you do not understand climate change, then you won't be able to innovate a climate smart technology. That is the reason you need to understand what is climate change and what is going to happen because of climate change. So these are the basic scenarios at different temperatures. What could happen at 1.5 degrees Celsius, global warming 2 degrees Celsius, 3 degrees Celsius, and how will the ecosystems will start collapsing. And due to climate change, the main reason what is happening is that the summers are getting more hotter, the colds are getting more colder, and the rains are getting more wetter. So you need technologies in order to make sure that you have correct technologies to battle heat stress, to battle the cold, the frost, which comes after cold waves. Then there are multiple reports, multiple cited reports where ice sheets are collapsing, basically the forests are burning, there is glacier collapse, and so on. And the main 2 reasons for this happening is illegal deforestation and rapidly melting ice caps. So something which is illegal, you cannot put an end to that. You cannot put a cap over it, and you cannot stop that. And that is the reason that why the ice caps are melting at a very alarming rate. And when there are so many problems around, so due to climate change, there is weather unpredictability, rains are happening at a different time, farmers struggling to make income because rains which used to happen in June, July, or October, September, they are shifting. They are having intense heat waves, they are having intense weathers, so there is lower crop yield. Then the nutrition density of
food which we get in market is also very poor because of rapid industrialization of food industry. Then food security, a global food security is one of the main challenges on which the Food Organization and Agriculture Organization is working. The UN is working, World Health Organization is also working, and a lot of people, a lot of scientific community are coming together to solve this grand challenge of basically to secure the food security in the era of climate change. Then agriculture pollution is increasing. There are less efficient practices which are being adopted by our farmers in India specifically. So when there is so many things, there are so many pain points, there's also an opportunity to innovate and to create something new. So drones, AI-based technologies, robotic harvesters, phytotron, indoor hydroponics, sensor technologies, controlled environment agriculture. So these are basically the future of agriculture, the climate smart agriculture or precision agriculture. And the pain point which we have decided to work on is the heat waves, which were witnessed last year, where record breaking temperatures were observed all across the globe and India, and such high temperatures were never recorded in history. So this is actually a waking call. This should be a waking call for all of us to think and move ahead towards a sustainable future. And due to these heat waves, which happened last year, a lot of farmers struggled because their productivity, the overall productivity, got reduced by somewhere around 40 to 45%. So if they are getting 10 quintal per hectare, they were only able to get 4 hectares or 5 hectares, oh sorry, 4 quintal or 5 quintal of wheat, where they used to get 10 quintal. So that is also affecting farmers' income. And with this problem in mind, we have created world's first climate smart fertilizer technology. So basically, this is a combination of humic acid, silicon, and some trace elements. And what we are trying to do basically is trying to regulate the transpiration rate of the plants when they are under heat wave or heat shock basically. So we have tested few results. Those results are currently confidential. We are currently planning to secure the intellectual property right for our innovation. But I would like to discuss few studies, scientific studies, which I have reported in my intellectual property. So this is a figure in which you can see the expression of various different genes. So basically these are differentially expressed genes, and this is the classification, a broad classification of these genes, and this is a subclassification of all these genes. So it was observed that after application of humic acid to the plants, these genes were upregulated. So, and these genes also have genes which are related with the stimulus or the stress conditions. So they were up regulated. That means that my plants were protected against the heat stress. This is another mechanism in which these are basically humic acid was applied when there was heat stress, and these are basically heat shock proteins. So there are certain proteins which is basically a part of plant mechanism, a defense mechanism to tackle against heat. So these proteins get upregulated in a heat stress. So after the application of humic acid, the genes upregulated, and these genes helped basically to make sure that the proteins, the normal proteins which are present in plant cell, basically they are not unfolded and they remain folded, so that the plant metabolism does not get disturbed. Then there was a study which is again done by Talha Mustafa in 2021. It is again published in Nature Scientific Reports. And over here, people applied silicon to the plants, and under heat stress, it is clearly visible that under heat stress as compared to ambient stress. After application of silicon, that is 2 millimolar silicon and 4 millimolar silicon, the plant responded in a positive manner. There was still a decrease in yield, but yes, there was definitely a increase in the plant yield. So basically, the two parameters which are presented over here is biological yield per plant and the grain yield per plant. So these both the literatures were reported by Nature scientific reports. Apart from that, I would like to tell you that why do you need to innovate to battle climate change? So this is for sure that no one is going to help you. Only the youth needs to make sure that they innovate technologies which are able to battle climate change. And the reason why you should innovate is basically you need to secure the future of the coming generations. Then you can easily make money. So entrepreneurs, this is what entrepreneurs do. So entrepreneur is basically a very tough journey, but still, it's when you get into the flow and when you start making some money, then is when you really start growing in the industry. Then these are basically few ideas which we are currently working on. Few are in prototyping stage, and basically this is the crux of the technologies which are needed to create a climate resilient infrastructure. So controlled environment agriculture. So I don't know how many of you are aware of, but there are geothermal based poly houses. So this is an innovative technology which could actually revolutionize the controlled environment agriculture. So there are very few people who are working on this technology, but this has great potential in future. Then microbes as bio control agents. So there are beneficial insects, there are beneficial microbes which can kill different pests in the field. That could be used as biopesticides. They are effective and technologies are coming in terms of biocontrol agents every day. Good seed technology. So you need to ensure that the seed you are sowing is of good quality. There are different parameters on which seeds are evaluated, and there's still a lot of research which is coming up. But technologies related to seed technologies, for example, disinfection technologies, the extraction techniques, have a huge potential in future. Then agriculture sensor technology. So agriculture sensors are recently introduced in science. So previously we didn't had a lot of difference in sensors, but as the world is moving towards a more data-driven economy, data has become the new oil, or some say that data has become new soil. So whatever is the matter, but we need sensors to collect that data, right? So it is very important that we have good sensors, reliable sensors, and which need to be produced basically to collect data. Then IoT-based data acquisition. So Internet of Things is becoming widely accessible to a lot of people with high connectivity. And in this particular approach, one needs to make sure that we have drones, satellites, and sensors all together at a one single platform. Because there are a lot of companies who are working on this particular idea, who are providing farm advisory to farmers, but they lack somewhere. Technology lacks somewhere. So we need to make sure that we integrate all these three platforms together. Then drone technologies. So in Africa, there are drones which evaluate the air wind, which evaluate the sunlight, and based on that data, they provide advisory to the African farmer that how much irrigation is needed today. So those models are coming up, which are really important and could really help a farmer in increasing its efficiency. Then regenerative agroforestry. So nobody talks about regenerative agroforestry now, but down the lane, 5 years, 7 years, regenerative agroforestry is going to be like a boom in industry, especially in India, because no matter what we are doing with the agriculture industry, forests are the lungs of our planet. So in the era of climate change, forests will be really helpful. And then indoor hydroponics. 2 minutes. Yes. Thank you. And with that, I would like to say just quickly say two things. First is, for the first time in the history of industrial revolution, science and technology has provided us with the possibility to help mankind transit into a sustainable and green economy. So this was never possible before, but now we have those resources and tools which can help us transit into a sustainable economy. Second is that there's one sure shot mantra for success in life, which I believe is that you need to work for 15 hours in a day for 5 years continuously. And you need to make sure that you dedicate towards education, you are curious towards science, and your time is dedicated towards hard work. Thank you.
Thank you, Dr. Rohan. Thank you very much. Now, number of questions, please. He's really open a list of solutions. So I think we should interact with him, try to find out the real answer. Last he concluded that 15 hours a day. How many of you want to work hard 15 hours a day? Not me, John. Any question? Yeah.
Can you please elaborate on regenerative agroforestry?
So basically, regenerative agroforestry is basically like a models. There are different models. So we need to make sure how much area you have, what area or what region you are, and then you need to make sure that there are different layers in forest. So there is one trees which grow tallest. Then there is a second type of tree which grows intermediate between those big trees, and then there is third, which are basically low-lying shrubs or crops. Okay, so and the best part about regenerative forestry is that the leaves which are fallen from the tall and the medium and the small, they gets converted into manure, which helps to forest to survive without even you basically can say that with minimal human intervention, the forest is able to survive itself, and it is still the soil remains very intact. It always remain high in humic acid, basically inorganic carbon, so which is really important for forest. So that is where the regenerative word comes. So you need to make sure that the ecology of a forest remains intact.
Okay, any other question? If not, I have small query.
Yes, sir.
What is the mechanism of evaluation of these kind of stresses, basically the internal heat stress you talk about?
Yes, sir.
With the help of humic acid as well as silicon. Carbon, I missed that.
So we that is a proprietary formulation. We have it.
No issue. But how this say if we are talking about silicon or humic acid, how they work? What is the mechanism?
So basically, humic acid, what it does, it acts on the heat shock proteins. So these are it upregulates the genes which help in formation of heat shock proteins. So it helps prevent the plant from the heat shock.
It has been there is plethora of literature where they have shown how to enhance the level of these kind of heat shock proteins as well as heat shock factors.
Yes, sir.
But nobody has proved with this kind of technology that we can enhance the productivity and production of high temperature.
Yes, sir. So we are not claiming that humic acid is going to do that, but silicon might be helped. So basically, silicon has a different way of providing the heat tolerance to a plant. So basically, silicon acts as a barrier. So silicon is similar to carbon. So it forms the surface, or basically the leaf surface. It's a leaf. So if you have a silicon over here, the harmful rays which are having high energy are not able to penetrate because of the silicon moiety. And the other reason is that the silicon provides the ability basically of osmoprotectant. You can say silicon provides the ability of to a plant which is called osmoprotectant. So it helps in basically osmosis regulation. Okay, and the third, the agents which we have not disclosed, so that is something which helps in transpiration. The third component of our recipe is basically that is helps in transpiration. So we are hypothesizing this, we are proposing this. That,
Thank you, Dr. Ross.
Thank you.
Very much. It's very happy. So next in line.
Now our next invited speaker is Dr. Muhammad Iqbal Khan from Department of Botany at Jamia Hamdard. He worked as postdoctoral fellow at the International Rice Research Institute, Philippines. His research interests lie in the area of plant stress physiology and plant systems biology, ranging from theory to conceptualization to implementation. He had been awarded and honored with the Young Scientist Platinum Jubilee Award, the Junior Scientist of the Award 2017, and recently he recognized as the highly cited research 2022 by Clarivate. Please, sir, join us.
Very good afternoon, sir. As you know, I'm working here, so most of the students sitting here. So I will start with my orientation, like why abiotic stress is important and why you need to take care with the plants, especially. So, okay. So today I will be orienting that how stress hormones, that is ABA and ethylene, and how the defense system of the plants can cope up with the abiotic stress tolerance. Especially, I will giving the examples of salt stress and like cadmium or arsenic, right? So as you know, we always talk about food security, nutrition security, climate change, zero hunger, sustainable development goals, so many terms, right? So what basically it needs? It needs some strategy to cope up with the abiotic stress, that is climate change. It may be the salinity, it may be heavy metal toxicity, it may be drought, submergence, anoxia conditions, high temperature. There are so many abiotic stresses. But I will be targeting salinization and some contamination of the metals. But they need attention to feed the people. The population is increasing day by day, and we know that everybody needs at the end of the day 2 times of the meal, right? In that way, we at Jamia Hamdard working on the SDGs programs. So salinization is increasing day by day. As quoted by 2050, it will be like 50% of the soils will be like salinity, right? And the contamination of water due to some anthropogenic activities, withering processes, fastings, the heavy metals are accumulating in the river and ultimately it's going to our agricultural land. So basically, we are at the edge of food scarcity. Food is producing very high. We have the high yielding varieties, high yielding and hybrid varieties, but still the population is rising and climate change is frequently happening. And it would be like food security plus nutrition security by 2050, and the population will be like 10 billion. We are talking about the 10 billion people feeding by 2030 or so on. So in plants, it will be like some mechanism which are going to tolerate those environments, and they will helping in the inducing the stress resilience. So I'm choosing the ethylene and ABA as a stress hormones, and how these stress hormones can help the plants to develop a resilience. So basically, we know that the basic ethylene or ABA will be enhancing due to the stress condition. And that's why they are known as the stress hormones. But how those stress hormones are very beneficial to the plants, and how these are going to be optimized through some mechanism or some supplementation or through the genetic engineering, right? So anaerobic stress, another oxidative stress trait is there. We have the ROS, RNS, and these are also like enemy of the plants. Okay, so we need some strategies to cope up with those stress hormones and those ROS or RNS production, which may be. So I chosen few phytohormones to tackle with these traits, the oxidative stress traits, elevated stress hormones, and among these, itself, ethylene, ABA, nitric oxide, and a new molecule that is known as GABA, and I synthesized few particles from the proteins of the wheat. So these are going to optimize how they are going to optimize the ROS concentration, these stress hormones, and how they are accelerating the defense system will be shown. Yes. So these plant hormones can control lot of mechanisms, lot of traits, including the yield. So let's talk about the ethylene first. Ethylene is basically in biphasic in nature. Biphasic means it can act as oxidant and reductant, but at the concentration of optimal level. So in plant physiology, it was reported in 2015 itself, that how oxidative stress ethylene is going to reduce through some elevators like salicylic acid, and how this salicylic acid is going to control your RNS, that is nitric oxide, and it becomes the photosynthetic performance with the high level of the ethylene concentration. How ethylene concentration is going to be optimizing the ethylene concentration, the nitric oxide generation, and ultimately how they are going to control the defense system, that is your sorbate pathway. And ultimately that salicylic acid is going to be regulated with some elements, that is nitrogen and ethylene, sulfur and ethylene, and the expression of the biosynthetic genes of ethylene is going to be hampered with the nitrogen supplementation. And ultimately, you can see that the relative expression of ethylene biosynthesis and gene ACS is going to perform all photosynthetic performance. Optimized ethylene is going to perform all photosynthetic processes. The another strategy was to control abscisic acid. How abscisic acid is going to be controlled, and in that purpose, I use some synthesis of nanoparticles, that is silver and nitrate, and we characterize that nanoparticles with a very high accumulation, and that nanoparticle was very prominent size of 27 to 33 nanometer. And these significantly modulated the stomatal conductance, the stomatal traits, and ultimately the stomatal frequency and electrolyte phase, all oxidative traits was controlled through the stress, through the ABA modulation. The another was that the AMP, and we targeted the nitric oxide control. So nitric oxide was controlled, that is RNS control, and the same stomatal traits was very prominent under the influence of these nanoparticles. You can see how they are influencing the oxidative traits and the shoot and root sodium and potassium ion under the salt stress. So we will be talking about the new molecule, that is GABA. GABA is basically a neurotransmitter in animal system, and very less investigation has been done on the GABA. But choosing the mutants, the biosynthesis of GABA, we observed that the phenotypic variations are very prominent. So after that, we selected the salinity and GABA treatment, and we found there is something which we can be explored. In that way, we studied the GABA with the salinity stress and observed that it influencing the nitric oxide and the stomatal traits and the scorbut pathway, and ultimately the plant dry mass and photosynthetic potential was protected through the modulation of nitric oxide. These are the very prominent results from the GABA treatment, that the cell death of the wheat plant was reduced, and the cellulose deposition was observed that they are going to be very reduced, and the superoxide localization was controlled through the GABA application. So these are some results at the yield stage. At the yield stage, you can observe that the phenotypic variations was very prominent, and these are going to be helped in maintaining the grain yield also, because GABA has a role to control carbon and metabolism ratio, and it is going to be help in your yield also. So these are the various phenotypic traits which are very prominent through the GABA supplementation, and these all have been published in Journal of Biotechnology already. So we tested how GABA is going to control other nutrients also. So we explored, as it was the salinity stress, so we explored the potassium with GABA, and that was found that the potassium supplement for GABA is very prominent as compared to the GABA itself, because in any cell consumption, you know, retention of potassium is very important trait to protect your yield traits also. So very prominent NaCl, GABA, and GABA plus, and so on. So all the visual traits are very clear that GABA and potassium is working very well. The same traits was observed under the stomatal conductance, and stomatal density, frequency, ratio, all were maintained. So you can take a message from that: optimizing stress hormones and optimizing the ROS and RNS can be a successful tool for the optimization of stress-induced oxidative stress, and that can be led by the various signaling molecules. The signaling molecules like GABA, the phytohormones like ethylene and ABA can be used. So this is my team. I really thankful to them. They are working very hard, and I thank Jamia Hamza, UGC, DST, and SERB, DBT for the funding my knowledge. Thank you so much.
Thank you, Dr. Ibalna, for the talk. Now, quick questions, quick queries, quick comments from the audience, from the students. No question means you understood everything. So, if nobody is asking, Dr. Khan, how the GABA is affecting the process of ROS scavenger system?
Yeah, actually, in detail, I did not go because there is a limitation of time. So basically, it is circulating the carbon insulation and nitrogen insulation. The expression of the nitric transport, the transmembrane pathways, enzymes, and genes are very expressive in the GABA. Because it is due to the GABA is a molecule which only control these two types of the metabolism. So these are all related to the yield and harvest generation.
So, is there any success story related with the
Yes, we have already.
Not from your group, means some other group. must
Yes,
be
yes,
working, so
yes, people are working.
Is there any commercialized product?
Yes, there is one group in Riken, Japan, they are working on tomato. So they found that the under tomato, they make a mutant for the Java synthesis, and they significantly found that the yield was reduced. That is no longer used for the Java. So it was well established. Now people are working on Java very frequently, but in India,
Yeah, I read somewhere that one commercialized product has been released by genome editing approach. This one can. So what is that particular enzyme which they target? Like what somebody
yeah, actually it's for Java.
Yeah, for Java.
It may be GAD.
GAD. Yeah. Okay. Thank you, Dr. Iqbal. Thank you very much.
Thank you, Dr. Iqbal sir, to pave a way for inhibiting stress and improving the defense system for environmental sustainability. I would like to request to our chairpersons to please felicitate Dr. Iqbal sir. Our next invited speaker is Dr. Tapan Kumar Mondal, is a principal scientist at NIPB New Delhi, working on abiotic stress tolerance of rice, on salinity and deep water stress tolerance, and also on tea. He has decoded the genome of Oryza cortata and tea along with their organelle genome. He is the recipient of many scientific awards, including the Fellow of Nasi Allahabad. He is the associated editor of many reputed journals also.
This is the pointer. So you like this. Yeah. You like to press it. Yeah. And then just forward. And this is backward. Okay. This pointer. Yeah. Thank you.
Good afternoon to all. On the outset, I'd like to thank the organizing committee, Professor Abdul. And there was a mistake, I think, in the technical program. It was written as a T genome sequence, but actually in the abstract I sent is this. So there may be some communication gap. I'll be talking on this plant. This Oryza quartata, and my topic is on wild relatives of rice. Actually, you know, the genus of Oryza is having 27 species. Out of that, Oryza quartata is one. So I'll be giving a little bit of introduction about this. Is it audible, no, from here? Yeah. So, you know, Oryza quartata is the only halophytic species among the 27 species of genus Oryza, and it grows in the coastal region of the Indian subcontinent, and it can grow up to the 40 EC of the ocean water. And earlier, when we started this work around 10 years now back, previously it was known as tetraploid with a genome of KKLL. But once you listen this lecture, you will see that there is a change on this, because we have found out that is not true, it is something else. To tell about this crop, this is highly salt-loving crop, and there are two reasons why I choose to work on this. Number one is that it is exclusively found in Indian subcontinent. I'll show you. And second thing, what attracts the scientists now and before also, it can grow in a salinity stress in which other normal cultivated crop are toxic. So there is a question that how it can survive under this high saline condition. You can see easily this crop is exclusively found in this Indian subcontinent, from the Andaman to this Gujarat, and in the world, in the Myanmar, Bangladesh, as well as in the Pakistan, Sri Lanka, this coastal region, it is found. So India is the major part where it is found. So that is what it is, an indigenous to India crop. And if you can see the EC value, it can grow in this region the EC value varies from the 20 EC to the 60 EC. So, what I want to say is that it is highly salt-loving plant or salt-tolerant plant found in the coastal region. Another property which is known about is that it is highly submergence tolerant. So these two things are there in this plant which attract us also to know what is the mechanism and how it works. So when we started this work, nothing was, not much, little information was there, not much known. Only few sequences was submitted, and there were two research institutes which is to work on this, is the Bose Institute as well as the Swaminathan Research Foundation. They were doing very excellent work, taking the individual gene on that. So in the beginning, we thought that we will going for the whole genome sequence to know what is there inside this. But with that, we started, and this is my outline of the talk. I'll be talking on this, but I give this lecture because in my lab, 3 of the students doing PhD, and incidentally, they are registered in the Jamia Hamdard. That is the reason I choose this topic here. And I'll be talking about the genome sequence of this, and then cytogenetics works, and little bit of the chemistry, and then RNA-6. In one area, we have failed, despite several tries with us as well as else, even in ERI, that there is a crossing between these two rice is not possible. And we have also find out what is the reason. There was a several attempts, and that is the major limitation for utilizing this crop as a donor for the salinity tolerance rice. So in the genome assembly, I'll be just, I keep on telling this topic, but today I'm going to tell you the updates on this. Well, before that, I would like to say NIPB in the morning time, my colleague Dr. Kishore has also given a lecture. We have done, this is the recent, this is a bit recent genome sequencing which has been made by the Indian scientists. Few crops has been also added after making this. But among this plant or the crop which has been done by the Indian scientists alone without any foreign collaboration, I will tell that this plant, Oryza quartata, is bit unique because of the polyploidization. It is a triploid now, but other rest of the more or less all the crops are diploid. So there is a problem with the genome sequence in the ploidy, higher ploidy of the plants. To start with the genome sequence, everybody, all of you know, we need to know the genome size because the cost, time, requirement for doing genome sequence depend upon the genome size. So we did the flow cytometry, and we have find out that the genome sequence is the 665 MB. Against the published literature in that time, it was reported that 1,200. Later on, we dig out the literature and we find out that that earlier estimation was based on the KMR, and which is not true. We have done number of times with 3 different samples collected from 3 different regions of India, and genome size is found more or less 665 MB. So with that, we have started this, and just for the student, for any genome sequencing project, you might be knowing, we take the DNA, make into the small pieces, and then we do the sequence at the ATGC level, because it is difficult to do the entire DNA of the cell to do at a single go. So we break into the small pieces, and when we break to the small pieces, then the next step is that we keep on trying adding them to make it bigger, as big as equivalent to a size of the chromosome, which in terms of bioinformatics we call as a pseudomolecule. So a ideal genome sequence in a very simple language will be one chromosome should represent one pseudomolecule with the same size. So we once we know the genome sequence size of this plant, then we started generating the data, and there are different type of data based on their size and chemistry. So we also did some different type of platform we use here. We first used generated the Illumina short read sequence, and then we try to assemble them, make the quantic. Quantic means then when the size of the assembled small pieces attached together. Then we put the Illumina parent data to make it bigger in size, so that the quantic becomes scaffold level 1. Once the scaffold level 1 was developed, then we put the nanopore, and then it becomes the scaffold level 2, that is the much bigger size. And after that, we fixed the genome assembled, and we published this paper that was a version 1. And what has happened after that, because that was a draft sequence, and draft sequence is not good because it is having a lot of gaps in the genome. So then we keep on working on this, and then later on we put the back sequence on this, and we make the scaffold bigger in the super scaffold size. The size has been increased. I'll show you the data. And after that, we have put the HiC data. HiC is these are the different type of platform. HiC is the assembled of the super scaffold into the chromosome level. When you put the HiC data, it should come all the parts, small parts of the DNA should join together and make the pseudomolecule, which should be equal to the n number of the chromosome of the cell. So we have done that, and now we have achieved up to the pseudomolecule level, but still there is some issue. We are working on that. So while working on this, we have used the Oxford nanopore sequence, which produced a very long sequence, and we have done the reference-based chloroplast genome sequence and decoded the chloroplast. Whereas we have used the short-read sequence and we have used make them the mitochondrial sequence, which we call as the de novo assembly of the mitochondrial sequence. See, this is the stat of this. First, the level 1 is this, which we published. Then we put the back library. Once we put the back library, scaffold number from 58,000 plus has reduced to the 12,000. And once on the black library, when we put the HiC data, the scaffold even has been reduced to the 124 number. So the entire small molecule has joined together and now come to the 124 number. But if you see closely, out of this 124 number, you can find out that 36 are in very large size. Out of that, 2 are still small, and rest 84 numbers are a very small size. They range from the kb size, that is 300 kb to 220 kb size. So this 36 pseudomolecule or the large molecule, they comprise of the 95.65% of the genome, indicating that they represent the chromosome. But we are still working on this because there are 2 pseudomolecules which are small in number. They do not represent the true chromosome. So we are working on this. So in future, so in near future, we'll be able to make the real size of the pseudomolecule. Now, this is just a vital stat of the genome. So our genome size was 665 MB, and we could achieve 570, and which represent the 85% genome. That means that 85% of the genome has been sequenced. Important stat here that the number of scaffold is 124. Out of that, 36 are large in size, and rest are the small unanchored size. And the percentage of the non-ATGC, which represent the gaps, is only 0.73%. And the total number of the repeats has been found nearly 20% of this genome. And when we put the RNA-seq data on this and to validate that, we find out the gene model is 33,000. That means that we have annotated 33,000 plus gene on this genome. So there is some measure also that how good your genome assembly. These are the standard practice called BASCO score. Previously it was a sigma also, and here it shows that BASCO score is 92.34%, indicating that genome is it's okay, it's good. Above anything on the 90 is good, and beyond 96 and above that is very good. And BASCO for the genome completeness is 97.08%. We also beat out the SSR because that is the need of the breeder, and we have found out more than 2 lakhs of the SSR marker. I'll give a small example that how we are using this. And we also find out there is a transcription factor. They are present here, but 4,000 plus genes in Oryza quartata distributed, which is called as a transcription factor, into 56 different transcription factor family. So important point is that when we find out, many people ask that what ultimately you got out of this genome. We have find out that there are 2,000 plus genes which is present in this genome, which is unannotated and not present in other. So from there we have picked up 11 genes which is highly upregulated under the salinity stress. We have done the qPCR and putting upon some of the selection criteria, we have finally chosen the 11 genes to characterize. I'll show one of the example today. This is the different repeat region which is present. Ultimately, this is the 20% repeat present in this genome. And one of the things which people are looking, and we are also looking, that who is the progenitor of this genome. So there is no clue because the ploidy level was a question. But when we compare, make an analysis with the 1:1 ratio of the orthologous gene, that is a standard for making the evolutionary study, we have found out the Oryza brachyantha is quite similar with the Oryza quartata. And you will be surprised to know, those who once knows the wild species of rice, they are morphologically very similar. And many a times there are other evidence also that they are very close. So we are suspecting that brachyantha could be one of the progenitor or the vice versa. But brachyantha belongs to the FF genome of the Oryza. We also did the synthetic analysis and we find out some unique percentage of this genome which is not present in rice, that represent 2.19% of our 85% genome assembly, and we suspect that these regions are very unique, maybe responsible for the salinity stress tolerant of this plant. This is the chloroplast genome we did, and we have also validated this through wet lab, the junction of the chloroplast genome. These are the strat of the chloroplast genome. I'm not going to details on this. Similarly, the mitochondrial genome, and then we published this paper with the first assembly. Now I'll tell you some of the application part we are doing. As I told you that 11 genes so far we have targeted based upon their expression and some other criteria. These are the criteria. So we have picked up them and we are doing transgenic validation of that. But doing any kind of qPCR analysis, you have to standardize the reference genome. That also we did and published. So I'll be giving one of the example of one gene. This gene is called OC1330. This is uncharacterized gene, but it has the 35% protein level similarity with the rice genome. Rice Arabidopsis genome. I'm sorry, but it is not present in the rice genome. And this, when we check this, it has the we have 1 hour and 6 hours. The qPCR analysis shows that it is upregulated under the salinity. We check their expression in different plant parts, find out that flag leaf as well as the roots is having high level of expression. We made the construct, made the transgenic, and you can see here that very clearly at 3 different levels of this gene, when we put in the Arabidopsis, we have tested, find out that wild type could not germinate that well, but the 3 different independent events, they have shown better germination under the salinity in 100 millimolar sodium chloride level. So you can see at seedling stage, this is the wild type, and this is the 3 different independent line. They are much robust, more tolerant with compared to the wild type. And this is the quantification of the root growth, and we have find out in the transgenic plant, they have not only the number of root, but the length of the root and the volume of the root are much higher than the control or wild type plant. And you can see this in the later seedling stage, when we put the 21 days seedling under the salinity stress, you can find out that the wild type was injured, whereas continuous putting the saline water in the transgenic line, they looks as compared as to the control plant, indicating that this gene works against the salinity. We have also tested different other abiotic stress and hormone condition, and we have find out that it is also tolerant to the ABA, because this is the wild type which could not germinate under the 0.1 millimolar ABA, whereas the transgenic line could do. And then it is also, this gene is also worked against the old tolerance. You can see easily that the wild type was the bleached, whereas the dying and the transgenic lines was there. Another fantastic result we got in the drought stress. You can see the control plant has been dried after recovery, whereas the transgenic plant was much more better than the wild type. So we do not know the reason, but then it could be there's a lot of crosstalks. So this gene maybe belong to the other stress as well. We also find out that it localized to the nucleus, so it is a transcription factor in nature. And then coming to the marker, I'll be giving one example. There are a lot of SSR marker we have generated, but ultimate aim is that to use these for transferring the gene, if we can make a cross for this. So we have done in collaboration with Rice Research Institute, Kotok, and we have done that. We have surveyed several SSR marker, and we find out that 2 of the SSR marker is specific to the Oryza quartata. Simple, this picture you can see lane number 18 to 23 are the different accession of Oryza quartata, and these are the different species of rice as well as other BB and FF genome of the wild rice species. And you can find out that this marker is very specific to the Oryza quartata, whereas it could not have any allele of among the other rice or wild rice species. This paper has also been published last year in Infitica. So the first part is over. Now I am going to the second part of this. As I told you, that this plant was very problematic for crossing. So people has made many failed attempts, including us. So we do not know the reason. The student who was working on this, I was telling her to make a cross in collaboration with the IRI genetics department. She also could not do after attempting the 2 years time. That's the point where we suspect that something is there in the cytological issue for which it is not there. Then we started looking at the cytological level, and as expected, we find out this plant is the triploid against the all published paper reported as a tetraploid. Now we have done the many sample now, because last year we published this paper and we found that in several collected sample around 4 cases of India and find out that the triploid in nature. And our suspect was also confirmed with flow cytometry and other evidence. You can see that the pollen viability, there is a high level of pollen sterility in this plant if compared to the rice, and the seeds formed here do not germinate. That is reported in the literature, but the reason is not reported. When we looked into it, we did not find any functional embryo on this. It's a stone-like structure we find. Another important feature we find out that the presence of the cranch anatomy. So you can see that there is a cranch anatomy present here, but then we could not till now experimentally prove that it has the C4 activity in this. We did the IRMS study, and then carbon dating study indicates that it is still in C3. With this, along with there are some other evidence that it is a C4. There is a high vein density. Then there is a differential chloroplast number between the bundle cell and the mesophyll cell, and also the chloroplast orientation, functional grana, all are reported in this publication. There are high ratio of bundle seed and mesophyll cell area, presence of typical cranch anatomy in that bundle seed cell which encircled in the vascular bundle, chloroplast of the bundle cell set contain rudimentary cycloploid. These are the typical symptoms that it is going to be it is having the C4 machinery. So carbon dating status, carbon discrimination study, but did not corroborated with the anatomical feature. It is showing that it is still in C4. But the experiment of the photosynthesis has been done in IRI this thing. So we do not do in the actual niche area. Therefore, we just concluded that it is towards evolving the C4 machinery. We have published this paper. And another feature we find out that we did the LCMS and find out one of the compound allantoin accumulation is very high in the root. We dig out the gene for that and we made the transgenic Arabidopsis, and we find out yes, uricase, which is the main rate limiting enzyme of this allantoin, is giving the high salinity tolerance in the Arabidopsis. And the other work we have done, the small RNA with this. One of the accidental finding in this, that we have developed a pipeline. If anybody is interested, I will explain more, which can detect more number of microRNA, non-coding RNA in this. And we have published that paper also, and we have taken the publicly available data. We have used our pipeline and the reported pipeline, and we have find out that if you use our pipeline, you can dig out from the same data more microRNA compared to the presently available software. So this is the finding. We have validated this through the wet lab, published this paper, and as I told you that we have failed in the breeding because, but we are now taking different measure. All this result has been mounted in a Voyager Quartata database, which has been there. If anybody is interested, I don't know whether it will be working not, sir. I'll take 1 more minute. Yeah. So this is the database which is having all details of this. Yeah, so the future work is that these are the future work that we are planning for the association studies, because we have an indication now it grows in different soil regime with a different soil tolerance, and also the degeneration protocol is under process. Maybe in future we'll be taking to the neo domestication, and of course the crossing will be made now using this as a female parent, because people over the years was using the donor parent. So these are the conclusion of my talk, that we have made the genome sequence, and then chloroplast and mitochondrial has been decoded. 11 gene we have been find out, although we are now making a robust approach to find out more gene. And then it has the C4 anatomy, although it is non-functional. And then you have find out several microRNA specific to this plant. And this is the repeat content is the lowest among the all this genome. We have written one book on this, and finally, thank you for this. And this is my lab members. 3 of the students they did PhD on this crop, and they are all from the Jamia Ahmedabad, including one student who is working on this bioinformatics analysis. So that's
Thank you. So now I request the audience, students, to interact with such a wonderful study he presented on Eliza. A beautiful plant, I suppose. So must be curious to know about many things. Even me too. But I'm waiting for you to complete and then I will come. No question? After lunch, okay. Dr. Kamal Mandal, first of all, I congratulate really wonderful work. You completely dissect this particular crop. Really good. So, I would like to know, being a plant physiologist, I'm really interested to know about the C4 characteristics. As you explained that psychologically you have dissected, but functionally you haven't approved till date. But apart from psychological evidences, is there any other evidence you got it that by which you can easily prove that this is a C4 crop?
This part is basically looked by a plant physiologist, Dr. Rakesh Pandey. So it's not in our domain area, but not psychologically, anatomically.
Who is this Rakesh Pandey?
Yeah. Yes.
So he hasn't done this kind of work.
No, but he was associated with some of the initial work on this, and in the publication. Yeah. So he was this thing, but anatomically you have find out, yes, the C4 is present. Now also there is an independent report, they also reported that it has been from the China, this. But apart from this physiological data, we are not having much data. We did the C13 study for this. So C13 data is showing that it is not active photosynthesis, but now with in your division only, we have planned to do some enzyme assay with a carbon dating, which is the major study with Dr. Bhupender Singh as well as Dr. Chinnu Sami for this.
That will be really good because first of all, we have to prove this is a C4. Only then we can understand the difference between C3 and C4.
Right.
As otherwise, people are taking a lot of advantage of making C4 nice plant.
Yeah, that is the thing. That is why there are 2 things. Just I want to tell. We have spent a lot of money to Bill Gates for making the C4 rice as a C4 plant. So if we can take out the gene at present through transgenic, that could be useful. But as you say, we have made a plan for doing the carbon isotope study of the major C4 enzyme.
Thank you very much.
Thank you so much, sir, and lots of congratulations for the journey of decoding the genome of Oryza cortata. I would request to our chairpersons to please felicitate Dr. Tapan Kumar Mondal, sir. Now we have 3 oral presentations from young researcher. First, I would request to Ms. Asna Khan from Jamia Millia Islamia to give your presentation.
Good afternoon, everyone. Am I audible to everyone? So, hi, I'm Asna Khan, PhD scholar for Jamia Millia Islamia. I'm here to present my work on deciphering the role of stress-regulated factors in imparting tolerance towards the dual stress of arsenic and submergence. So, talking about the plant model that I've chosen is the rice. Rice is the major staple crop for a lot of people in the world. It is a semi-aquatic plant. And talking about its productivity, continent Asia and Africa is the major producer and consumers of rice plants. In India, almost all the states are rice-growing states except for Rajasthan. One of the major stress that rice productivity faces is arsenic. Arsenic is found to be a noxious metalloid which gets transported into the brain, and upon consumption, it causes a lot of poisoning to human beings. All over the world, continent Asia and Europe are the major reservoirs of arsenic. In India, all of the rice producing states are the reservoirs of arsenic. Arsenic is majorly generated either through anthropogenic or geogenic activities. It is found in two different states as inorganic and organic. So, talking about the effects arsenic has on human body, it majorly damages nervous system, cardiovascular system, respiratory system, and developmental processes in human beings. Another stress that the rice plants encounter is flood. As we all know, excess of everything is bad. Being a semi-aquatic plant, it requires a little amount of water, more little amount of water than what is required by the other plants. But when high amount of water is being encountered, the plants undergoes flooding. So there are three types of flooding that has been characterized. When the plant's root system is under the water, it is called as waterlogging. When half of the plant system is under the water, it's partial submergence, and when whole of the plant is under the water, that's complete submergence. So there are two types of strategies that the rice plant has opted in order to confront this stress. One is the escape strategy, where the rice plant elongates its shoots in order to come out of the plant, which further leads to its recovery. The other stress response that the rice plant has encountered is the questing strategy, where the metabolism has been restricted. The plant doesn't elongate its shoots. It remains of that height. And when the flux recedes, the plant elongates its shoot and do more production and growth. So the major rice producing region of India, as we know, is the Ganga-Brahmaputra basin. This major basin is being acknowledged with flood every year and is also the same area which has arsenic contamination. So this major region is what I am focused on about my study. So there is an ongoing project that has been done in our lab. We procured several varieties from this region after the flood has been receded. So the varieties which have survived the floods were procured. These were subjected to elemental analysis, and after elemental analysis, we get to know that there is a variety that is called as mini Masuri, which showed very less arsenic translocation to the grains. So this variety was taken further, and from this field to lab journey of this variety, I have been performing the screening of arsenic concentration in the lab. The screening for submergence duration was done. I analyzed the growth-related parameters and survival rate. Estimation of pigment, protein, and stress markers was done along with the arsenic content. So talking about the arsenic concentration first, we screened three different concentrations of arsenic, 100 micromolar, 150, and 200. However, not much difference was observed. But yeah, a slight reduction in plant height, a slight reduction in seed germination was observed. So taking that as the point, we chose 150 micromolar as the concentration for arsenic. That also mimicked the concentration that was found in Balia region. Talking about the second screening that was performed was submergence. The submergence durations were characterized as 3, 5, 7, 10, 12, and 14 days. As we all can see, the plant height increased in the 5 days duration, whereas it kept on elongated up to 14. The new leaves has been generated, the older leaves have turned brown. That marked as the survival strategy that this rice variety has opted, that is the escape strategy. So keeping that in mind, we took two sets of plants. One I characterized as underwater and the other I characterized as upper water. In the underwater plants set, the plant height was not increased, it was still under the water facing the submergence stress. And the other set that I chose was the upper water plant set, where the plant height increased, the water level, and some major amount of the leaf tips of the plants came out. So in order to confirm its tolerance and resistance mechanism, we studied the internode elongation and survival rate. For internode elongation, as we all can see, compared to the control plant, it's not working. Okay. Thank you. So yeah, compared to the control plant, this third leaf has emerged even more with 34 millimeter internode elongation, as we can observe in the submergence duration. And compared to the arsenic treated plants, this same plant has shown a 42 millimeter increase in the internode elongation. The increase in the internode elongation here is more compared to the controlled submerged plants because of this height difference that has been observed previously due to arsenic treatment. Then coming out towards the reaeration that we have provided after the 7-day submergence period, 7 days of reaeration was provided. After that, we observed the generation of new leaves. The fourth leaf started coming out, and these orange arrows indicate the browning or the thinning of the previous leaves. So, talking about the morphophysiological parameters that we studied, the shoot length and root length initially decreased when the plants were submerged, but with the advent of time, the shoot and root length was found to increase. Number of lateral roots also increased when the plants came out of the water, and the water content that was found in shoot and root were also found to increase. Compared to arsenic, everything decreased initially, but yeah, with the advent of time, a little increase in arsenic-treated plant was also found. Talking about the photosynthetic pigment analysis, the chlorophyll content decreased when the plants were submerged in the water, but when the third leaf started coming out of the water, the chlorophyll content also increased. And the arsenic content was not, chlorophyll content in arsenic treated plant was not found to be much difference because of this tolerance mechanism that this variety must have shown. Similar trend was found in protein content. This protein content also decreased initially when the plants were submerged, but with the advent of time, with the coming out and reemergence of the leaves out of the water level, this protein content also increased. Another one very fascinating thing that we came to know was the protein content in root. In root, the protein content kept on increasing in submergence compared to the control and arsenic treated plants. This could be due to the production of anaerobic proteins that the plants must have encountered in order to protect itself. Talking about the stress markers, I took MDA concentration and H2O2 concentration, MDA being a lipid peroxidation indicator and H2O2 being a ROS damage indicator, were found to increase considerably under the submergence stress when they were under the water. But when the plants, when the leaves started coming out of the water, this MDA content also decreased. Similar trend was found in H2O2 content as well. Now, the stress-specific amino acids, which are proline and cysteine, these are the scavengers of the ROS or the oxidative damage that the plant has encountered with studies. Similarly, the same trend, increase in proline concentration when they were under the water was observed, and the same trend continued even when they came out of the water, maybe to facilitate that this protection mechanism or the scavenging mechanism of ROS has been, taken about till 7 days. So since these are, I was studying about the submerged plants. So one, regulation study that the submerged plants encounter is of sugar production or metabolism. So in that, I tried to study this anaerobic respiration enzymes, which are a major indicator of underwater submergence. So when this pyruvate is formed, instead of going for the TC cycle, it undergoes this lactate fermentation or the ethanolic fermentation in order to provide energy to the plants. So this pyruvate decarboxylase enzyme was analyzed, whose activity was found to increase when the plants were under the water. But with the advent of time, when the leaves started emerging out, the activity of pyruvate decarboxylase also decreased. Similarly, the activity of alcohol dehydrogenase was also observed. The similar trend was found. However, the activity of alcohol dehydrogenase was more compared to pyruvate decarboxylase, indicating that the cellular toxic acetaldehyde that was produced because of pyruvate decarboxylase was scavenged perfectly in order to not make it toxic for plant anymore. So this alcohol activity was also found in arsenic stress plant, which is an indicator that the limited level of alcohol dehydrogenase activity in arsenic stress plant indicate the tolerance behavior towards arsenic as well. Now talking about the arsenic content that was analyzed through ICP-MS, the shoot and root samples were subjected to ICP-MS, and we found that arsenic translocation in shoot was very less compared to the root. The same result was obtained when we subjected the samples procured from the Balia region to the elemental analysis. So this also validated our studies that the arsenic was very less translocated, providing the plants with the tolerance. Here, as we can see, with the advent of time, the arsenic concentration has also decreased when the plants were above the water. So concluding it, we have provided enough evidence to support the tolerance behavior of the plant towards arsenic and submergence, indicating it as a climate smart variety, which can be used further in the places, in the further future conditions when the plant or the areas will be affected by floods and arsenic. So major points that came out from our study was alcohol dehydrogenase, which was a very important enzyme signifying the tolerance behavior of the plants towards both arsenic and submergence. Concentration of proteins in the roots increased, which signified the production of anaerobic proteins under the stress condition. That's my study. Thanks.
Any question?
Yeah. Anaerobic, this is not germination. I have already germinated the plant for 8 days. This was anaerobic respiration enzyme analysis. So when the plant undergoes submergence, it starts to, the respiration has been switched from aerobic to anaerobic.
Yeah, that's fine. But I just wanted to know how you are.
The treatment, I grew the plant for 8 days. 8 days, up till the vegetative stage, when there were 2 leaves of that plant. Fine. I then submerged it in this transparent container. Yeah.
Plants are 4 weeks of their growth.
Yeah.
And the height of the deep water is around 1 meter and more than that.
Yeah.
So what you are talking, I think it is anaerobic. These plants, what you are showing is long-acting.
Yeah.
They are anaerobic germination tolerant. So that is it.
So I took it as like the germination has already happened, right?
Even that
G.
So AG tolerant genes, even if you pre-germinate them, all you put it under the water, they will start keeping going.
Yeah.
But that is a totally entirely different genetic mechanism than the
Yes, sir.
Therefore, I was looking.
Okay. So, but we analyzed it through reaeration process as well. We reaerated the plants for 7 days. And after reaeration also we observed the growth in the plants, their plant height. Yeah.
Before the means your plant height will be more than 1 meter tall, even if it is 2 meter tall, we have some.
But since I took the small duration, I took 3 days submergence and 7.
The deep water tolerance, they are susceptible to the water inundated in the initial stage. It is reported as rate of nitrogen. So if it is germinating below the water in the initial stage, so it's not a deep water mechanism. Then it comes under the age.
Okay.
Other person wants to. I have 3 queries. First one is that whatever the data you presented is on the fresh weight basis.
Fresh weight, yeah.
Your whole experiment is on stress.
Yeah.
Stress experiment should not be expressed on the fresh weight. It should be dry weight or protein.
So dry weight we have estimated arsenic content.
Arsenic content on it. That is the only part you have. Other means any kind of estimation I have seen you express on this.
Fresh weight, yeah.
So you convert these data into dry weight. Possible.
Okay.
Number one. Number two, you showed the complete means the on-demand productivity and production of life. Washing productivity. What I would like to know from you, what is the India rice productivity and production?
India, like there's a high scale productivity in India and consumption as well. So I'm not,
Another part is you showed one of the slides where you showed the strategy for surviving under any kind of critical situation. One is there are two kinds of strategy you showed.
Escape and talk.
Escape, another is resistance, right? So what kind of hormones are involved in that? That is a tricky word.
Yeah.
So that's the reason I'm asking.
Yeah, sure. I have actually done the hormone analysis, but the results are awaited. So for the escape strategy, gibberellic acid is the main hormone that is involved in it. Right? And for the question strategy, it's ethylene. Right. So
no need to explain. I wanted to know that whether it's
I have actually done that, but the results are awaited. Thank you.
Thank you, Asna. Now our next presenter is Narjis Sabah Khatun from Jamia Millia Islamia.
Good afternoon, everyone. I, Narjis Sabah Khatun, work as a PhD scholar at Jamia Millia Islamia, New Delhi, under the supervision of Dr. Neetu Gupta, Department of Biotechnology. Today, the topic of my presentation is deciphering the regulation of nutrient use efficiency in wheat cultivar under drought and arsenic stress. So, moving to the introduction. Wheat is the second most important dietary intake grain after rice and the most important internationally traded food crop. It has lots of health benefits, like it helps in prevention of neurodegenerations, strengthen body immunity, reduces risk of stroke, breast cancer, cholesterol, type 2 diabetes, colorectal cancer, coronary heart diseases. Moving to the next slide, this is the overview of plant mechanism influenced by drought. Due to global warming and rain-fed conditions, water shortage is one of the major abiotic stress threatening crop production and yield globally. So drought stress seriously affect plant owing to production of reactive oxygen species, due to which it obstruct ATP synthesis, stomatal closure, ultimately decline in photosynthesis, due to which crop production decreases. So we have another toxic metalloid, that is arsenic, which is present in earth crust. It is released into the environment by two processes, that is natural and anthropogenic processes. It has two forms, that is inorganic form and organic form. Organic form are most prevalent in seawater and are less toxic to humans and plant, whereas this inorganic form is most prevalent in soil and are highly toxic to humans and plant. Arsenic exposures and toxicity in humans. It's a known carcinogen and mutagen that can lead to both acute and chronic adverse health effects. So main route of arsenic exposure in humans is via drinking water or via food. Arsenic severely affect humans as well. They causes cancer. It also severely affected plants, reduces grain quality, hinder nutrient uptake, ultimately reduced in plant productivity. So this is the drought and arsenic co-contaminated area throughout the world. The combined stress of drought and arsenic has been found worldwide, mainly in USA, Mexico, Argentina, Nepal, China, Thailand, India, and Bangladesh. And this is the national status of wheat cultivation and drought and arsenic prone areas. Comparing these two images, we can come to the conclusion that the wheat producing areas, major wheat producing areas like Uttar Pradesh, Delhi, Haryana, Madhya Pradesh are also the areas produces approximately 10 to 30 million tons of wheat are also the areas which are severely affected by the combined effect of drought and arsenic stress. So therefore, some better crop management practice is required to cope with it. For that, we have nutrient use efficiency. It actually measures the ability of the plant to take up nutrient and use them to produce biomass. So the question arises where, why, and when nutrient use efficiency is required. So it is required in all environmental conditions where yield is required, because nutrient use efficiency is directly proportional to crop yield. So when nutrient use efficiency is required to minimize nutrient loss, maximize nutrient uptake, and reduce environmental pollution, and why it is required for the abiotic stress improvement in crop. So farmers are highly benefited from producing highly nutritious crop, but it is not easy and take diligence. So abiotic stress reduces nutrient inside the soil, due to which soil becomes depleted. If this soil is not if this soil is not restored properly, then it leads to a reduction in crop plant productivity. So for that, a supplementation of nutrient help in restoring this depleted soil and help the plants for their growth and production. So for that, in my study, I mainly worked with nitrogen and phosphorus, because these are the primary macronutrients for their growth and development. So the question arises, why nitrogen and phosphorus? In my study, I chose nitrogen and phosphorus to understand the regulation of nutrient use efficiency in wheat and how it regulates photosynthesis under the combined stress in the presence of nitrogen and phosphorus. And why photosynthesis? Because nutrient like nitrogen and phosphorus taken up the plant is assimilated for their growth and enhancement of the biomass, and which is regulated by sugar production via photosynthesis. So this is the role of this is the pathway and the role of nitrogen and phosphorus to regulate photosynthesis. Mainly, nitrogen and phosphorus relies on photosynthesis, and photosynthesis mainly relies on nitrogen and phosphorus. Inside the soil, nitrogen are present in two different forms, that is nitrate and ammonia, and phosphorus is present in organic and inorganic forms. Inorganic phosphorus is taken up by the plant, and this nitrate is taken up by the nitrate and ammonia is taken up by the plant via transporter, and inside the plant this nitrate is converted into nitrite, and then this nitrate is converted into ammonia with the help of enzyme nitrite reductase, and then finally this ammonia comes and combines with the carbon skeleton, which is produced by photosynthesis, and they helps in the formation of amino acid, which is used for the plant growth. And also there is another role of nitrogen. They help leaves to expand their area so that they can trap more solar energy during the process of photosynthesis. And phosphorus helps in the production of ATP, and this ATP is used as energy in the process of photosynthesis. So these are the methodology. So firstly, I did morphological and physiological analysis, and then I did biological analysis, and then I did nitrogen acquisition related analysis. Firstly, I did the screening of diammonium phosphate for the source of nitrogen and phosphorus. The seeds were germinated in dark for 2 days, and then these pods are transferred to culture room. For cultural room, and each pod is irrigated every other day with different concentration of diammonium phosphate, that is 10, 20, 30, 40 micromolar, and it was found that 30 micromolar concentration diammonium phosphate was found to be a suitable concentration for the source of nitrogen and phosphorus. Then I did PEG screening for the induction of drought. Here each pot is irrigated every other day with different concentrations of PEG. that is 5, 10, 15, 20%. And it was found that 15% PEG was found to be 50% lethal concentration hampering the plant growth. So 15% PEG was chosen as the final concentration for the induction of drought. Then I did screening of arsenic for the induction of metalloid stress. Here, each pot is irradiated every other day with different concentrations of arsenic, that is 100 micromolar, 150 and 200. And it was found that 150 micromolar was found to be a suitable concentration, was found to be a 50% lethal concentration hampering plant growth, so for the induction of metalloid stress. Then I did the composition of modified Hughlin media and used to produce nitrogen phosphorus deficient media. Here, what I did, I modified this NP deficit Hughlin media by replacing calcium nitrate, potassium nitrate, and potassium phosphate by potassium chloride, calcium chloride, and potassium sulfate. Then the question arises, why NP deficit media is generated. The motive behind this NP to find out, to understand the regulation of nutrient use efficiency in the absence of nitrogen and phosphorus under the combined stress, and secondly, to find out how this nutrient use efficiency regulated photosynthesis in the absence of nitrogen. This is the experimental design here, or experimental design comprises of 6 different treatments, that is control arsenic, drought plus arsenic with deficient NP, then drought plus arsenic, and drought plus arsenic with sufficient NP for a period of 7 day and 15 day. Then here it was observed that recovery of plant growth, it was observed that recovery of plant growth occurs under drought plus arsenic with supplementation of sufficient nitrogen and phosphorus as compared to all the other stresses, and whereas there is decline in the plant height under drought plus arsenic with deficient NP for both 15 and 7 days. Similar results were found in germination parameters. Here you can see that the germination energy is higher in drought plus arsenic with supplementation of nitrogen and phosphorus. So then we did photosynthetic analysis. Here we did chlorophyll and carotenoid contents. These are the pigments. These are the pigments which helps in absorption and absorption of energy and transfer of energy in the process of photosynthesis. So here we can see that it was observed that at both 7 and 15 day, it was observed that under drought plus arsenic with sufficient NP has higher chlorophyll and carotenoid content, whereas at under drought plus arsenic with deficient NP has lower chlorophyll and carotenoid content. So the reduction in chlorophyll content is maybe due to chlorophyllase activity, and sufficient nitrogen phosphorus enhance it, as chlorophyll molecule contain nitrogen, making this element an important factor in the development of photosynthetic apparatus. So then we did total protein content. Here here it was observed that with the passing of time, it was found that under drought plus arsenic at 7 day, protein accumulation is lower as compared to arsenic alone. But with the passing of time, it was found that under 15 day, under long exposure, it was found that drought plus arsenic accumulated more protein as compared to arsenic alone. This might be due to some mechanisms ignited due to drought, which is also helping to face arsenic stress. Then we did oxidative stress marker. Here we estimated H2O2 and MDA content. H2O2 is a marker for ROS production, and MDA content is a marker for lipid peroxidation. And it was found that under drought stress condition, the H2O2 and MDA content was maximum at both 7 day and 15 day. And this increase due to and it was also found that under arsenic stress, H2O2 and MDA content was least. This is due to increase in ROS production. So it was also found that addition of nitrogen and phosphorus decline H2O2 content, which might be due to stimulation of reactive oxygen species scavengers to combat stress. So then we did stress modulator. Here you can see that proline and cysteine content at both 7-day and 15-day. Drought contain more proline and cysteine. This increased cysteine and proline, actually proline is a osmoprotectant, and cysteine worked as antioxidant defense. Antioxidant, it has an important role in antioxidant defense mechanism. So increase in cysteine and proline content was found in drought stress depict a tolerance mechanism in plant, whereas it was also observed that under drought plus arsenic with deficient NP content less proline and cysteine compared to drought plus arsenic with sufficient nitrogen and phosphorus, signifies the contribution of NP in stimulating antioxidants. Then we also did non-enzymatic antioxidant. Here we estimated anthocyanin and flavonoid contents. These are the non-enzymatic antioxidants, and it was found that drought stress contains more anthocyanin and flavonoid as compared to all the other stresses. So this increase in anthocyanin and flavonoid might help in reduction of H2O2 with increased level of non-enzymatic antioxidant. Plant showed enhanced tolerance to drought. Then we did nitrogen acquisition related analysis. Here we estimated NR activity and NIR activity. So in this NR activity, this helps in conversion of NO3 negative to NO2 negative. And then this NO2 negative converted this nitrate into ammonia, because nitrate is highly toxic for plant and causes mutation inside the plant, due to which crop production decreases. So then we did, it was found that it was found that under arsenic NR and NIR activity decreases as compared to drought. It is possible, only 2 minutes, it might be possible that arsenic react at its active site, resulting in the inhibition of its activity. So this is the conclusion. From my study, it was found that the decline in plant growth under combined drought and arsenic stress is mainly due to significant reductions in photosynthetic pigments, accumulation of oxidative stress markers, which highlights the adverse impact of coupled stress on plant. But however, better performance of plant was observed during long exposure of drought and arsenic stress than arsenic alone stress. And it was also found that NP supplementation appeared as a promising approach in relieving the plant from combined stress of drought and arsenic. However, we still need to figure out the regulation of photosynthesis by nutrient use efficiency, and I'm still working on phosphorus metabolism also. So thank you for your patience.
Any question? Indeed. Your expression of data is basically on the fresh rate basis again.
Yes, sir.
So you converted your data will be changed. Okay, I assure you. Okay. So you are PhD?
Yes, sir.
So please discuss with your chairperson. I think he or she will understand it better. And if you change it, your result will be changed. I'm sure of it. Number one. Number two, the pen you use 15%. Right? So what was the water potential of that particular factory?
Actually, sir, I grew my plants in soil, right, not in water. Then I
showed that treatment of 15%.
Yeah, but I grew my plants in soil, right? Then I, supplemented with.
Right. So, but have you measured the water potential or the water status of the plant?
Sorry, sir?
Water status of the plant? Have you measured it?
No, sir.
So, how you can say these are under stress condition, drought stress condition?
I measured. I already screened that. With different concentration of PEG, and then find out on the basis of plant height.
That is indirect method basically. So you don't have, and okay, it's fine. Third one, if any kind of plant which is showing high photosynthesis rate, do you expect high productivity or high yield in those plants? If you have high photosynthesis rate in one wheat plant, okay. So do you expect high yield or high productivity of that particular plant?
Yeah, I expected.
But it is not happening. That's the reason. So maybe after photosynthesis, there are many limiting factors which is governing or which is regulating the total yield of the. That's what I wanted to know from you. If you know it, then you can take it. So thank you. Thank you very much for the presentation. So I think this was the last presentation for this session. I think organizer will tell us what to do now.
So now this is the time for felicitation of our chairpersons. I would request to Dr. Kishore Gaikwad sir to please felicitate Dr. Ajay Arora sir. Now I request to Dr. Ajay Arora sir to please felicitate Dr. Kishore Gaikwad sir. And both of you deserve it, so you got to. By wrapping up this theme, now we are entering in next theme, role of biotechnology in pharmaceuticals and nutraceuticals for human health, as pharma and nutraceutical approaches provide a food plus medicine combo in the market of sustainable development. I would like to request to chair this session Dr. Usha Kiran. She has done PhD in biotechnology from Jamia Hamdard with specialization in bioinformatics. Her research is focused on signaling modulators used by plant cells to evoke protective cellular responses. She is involved in several training and dissemination of knowledge related to bioinformatics. Through her initiatives on Varnicia technology, she has been published in national and international research in national and international journals. She also has 3 books to her credits on application of biotechnology in plant science. Please, ma'am, join us to chair this session. In this session, we have 2 oral presentations from the young scientists. First, I would like to request to Ms. Aisha Waheed from Jamia Hamdard to present her oral presentation.
A very good evening to all of you. A very good evening to all of you. Good evening, ma'am. My dear colleagues, Aisha Waheed, I am from Department of Pharmaceutics, School of Pharmaceutical Education and Research. I am a research scholar, ICMR SRF. The title of my presentation is Tailoring of QBD-driven epigenin-loaded cubosomes, Morphology, Penetration, and Cytotoxicity Analysis. So these are some of the snippets from my main research work. So coming on to the introduction part, epigenin, I have chosen the drug epigenin. It is a polyphenolic compound with countless nutritional characteristics. Currently, it is used in market as a nutraceutical and is being researched as a phytopharmaceutical in various diseases. Epigenin possesses remarkable anti-inflammatory and antioxidant properties.
From the biopharmaceutical point of view, it has low oral bioavailability, which minimizes its therapeutic window in various diseases. So in order to maximize its bioavailability, we chose the encapsulate our drug in cubosomes. These are the nanostructured liquid crystalline particles with highly ordered mesophasic structures. These cubosomes are the nanoarchitectonics, which consist both hydrophilic and lipophilic domains, which gives us the edge so that we can incorporate both hydrophilic and lipophilic drugs at the same time. It also provides enhanced drug permeation through various barriers within our body. So coming on to the formulation development, we first assess the various parameters that will eventually affect the final epigenetic loaded cubosomes. There are 4 parameters which we assessed. First is the material, second equipment, measurements, and process parameters. Under material, we assessed various the formulation parameters, which like basically ingredients which we use during formulation development. These include the solubility of drug, the log P molecular weight of the drug, the lipid which we used, surfactant, and the aqueous media. Equipment includes the lyophilizer, sonicator, and magnetic stirrer, which we utilized during our formulation development. The measurements include entrapment efficiency, particle size, and PDI. And the process parameters is the speed with which we formulated our formulation at the temperature and the stirring time of our magnetic stirrer. Coming on to the formulation development and its optimization, while formulating, we chose the approach of bottom-up technique. In this, we used lipid phase. The lipid phase used was glyceryl monooleate, and the aqueous phase was distilled water, and we utilized hydrotrope, which is an ethanol. What ethanol does, it liquefies the lipid phase, so it easily incorporated into the aqueous phase. The aqueous phase already consists of poloxamer 407, which is a surfactant. Then we drop by drop add our lipid phase into the aqueous phase under constant stirring with low energy input, and the cubosomes are formulated. And then we sonicate our cubosomes in order to get smaller particle size. Then we utilized box Bengen design with 3 factors and 2 levels. We chose our independent variables, glyceryl monooleate, poloxamer 407 in the percentage, with low and high levels of 3.5 to 4.5% and poloxamer of 0.5 to 1.5%. The sonication time ranged from 2 to 5 minutes. The dependent variables include vesicle size, PDI, and percentage entrapment efficiency. These are the effect of independent variables on various dependent variables. The first A, B, and C graphs represents the effect on particle size. As we can see from the first A graph, there is a significant increase in the particle size as the amount of lipid increases. So glyceryl monooleate has a positive effect on particle size. Similar with the poloxamer, it is a surfactant. So what it does, it reduces the particle size. When we considered sonication time in particle size, initially the particle size decreased due to the energy input in the particles, then it increased due to the aggregation, might be due to the aggregation, this is our speculation. First, the trend is like first it decreases the particle size, then it increases the particle size. Then in PDI and entrapment efficiency, a similar trend was observed. Glyceryl monooleate had a positive effect on PDI and entrapment efficiency. Poloxamer had a negative effect, and the sonication also decreased the particle size and PDI. Then using point prediction method, we predicted the values of glyceryl monooleate, poloxamer, and sonication time as 285 nanometers of particle size, 0.17 nanometer of PDI, and 83% of 83.8% of entrapment efficiency. When we formulated this formulation in the ratio provided by the software as 4:1:3.3, the experimental value came out to be 287.7 nanometer of particle size, the PDI of 0.15, and the entrapment efficiency of 80%, which is very near to the predicted value. The first image represents the particle size, which is 287.7 nanometers. It is a single sharp peak, which means that we have particles very close to 287.7 nanometers, and the PDI of 1.52, that means then that a formulation is monodisperse. The entrapment efficiency came out to be 80%. The second image represents the zeta potential, which is minus 32.3 millivolts, which shows that the formulation was stable for a longer period of time. Then we conducted optical microscopy. I circled two in the image. These represents the cubic images that were formed during in a formulation. These are concealed cubic images. That means a drug was successfully entrapped and there was no leakage of the drug. Then we performed X-ray diffraction. The first image is of the pure drug epigenin. The second graph is of epigenin-loaded cubosomes. So in the first image, the characteristic peaks of epigenin at various diffraction angles of 7, 10, 11, 14, and 16 degrees. Some peaks was also observed between 20 to 30 degrees. Then when we see in the second image, in epigenin-loaded cubosomes, we definitely see a diminished characteristic peaks indicating that the drug was completely dispersed in a non-crystalline manner. Although the peaks were very sharp, which means that our cubosomes penetrated up to 54.9 micrometers, which indicates that our formulation has superior penetrating potential and which can target deeper skin layer diseases. Then we conducted cell cytotoxicity assay with this on B16F10 melanoma cell lines. The percentage cytotoxicity of placebo cubosomes, epigenin suspension, and epigenin-loaded cubosomes were determined at various increasing concentrations of 12.5, 25, 50, and 100 micrometers. As we can see from the graph, a dose-dependent cytotoxic effect was observed with epigenin-loaded cubosomes. We also conducted IC50 value. The epigenin-loaded cubosomes and epigenin suspension came out, IC50 value came out to be 45.74 micrometers, whereas with epigenin suspension was 85.39 micrometers respectively. The conclusion was for my slides were the epigenin was formulated into cubosome in order to maximize the therapeutic window and to increase the bioavailability. The cubosomes were prepared using quality by design approach, including box Penkin design. The optimized cubosomes were characterized using optical microscopy and X-ray diffraction studies. The skin penetration analysis revealed higher skin potential of epigenin cubosomes, and a dose-dependent cytotoxic effect was observed in B16F10 cell lines. Thank you. Yes. No, it's not genetically modified. It is a liquid lipid. Basically, we formulate. This is basically a base material for our formulation. It is a liquid lipid which entraps our drug. This is glyceryl monooleate. This is not. Yes, this is something else. This is a lipid which entraps our drug molecule. Optical microscopy was conducted by rhodamine-loaded nano formulation. We, you can say the optimized formulation. We, in place of drug, we loaded rhodamine and see that the drug, the formulation, the cubic particles were properly formed or not. Thank you.
Thank you, Aisha. Now, next presenter is Zoya Malik.
ये पॉकेट में रखना है। ये माइक का है, इसको रखो पॉकेट में। हाथ में रख लो, कोई दिक्कत नहीं। ये फॉरवर्ड है, ये फॉरवर्ड हो, ये फॉरवर्ड हो। स्लाइड चेंज कर लेंगे। ये बैकग्राउंड है।
अच्छा ये रिवर्स है। Good evening all. I'm sorry. I'm Zoya Malik. My presentation topic is enhanced anti-tumor activity of endographite loaded nanoparticles in combination with a marketed drug in vivo. As we all know that chemotherapeutic drugs for cancer treatment have some side effects such as cardiotoxicity, loss of taste receptors, ovarian failure. So in order to minimize the side effects of chemotherapeutic drugs, we can combine it with a natural compound so that the anti-cancer activity is enhanced and side effects are reduced and simultaneously enhancing the immune system. So why to switch over to herbal approach? Chemotherapeutic drugs in the market have severe side effects and are considered as less toxic than, herbal compounds are considered as less toxic than synthetic drugs and long history of use in the treatment of cancer. These are cost effective and less expensive than the synthetic drugs. The focus is to work upon the body's natural immunity and ability to take the disease and destroy cancer cells. So we have chosen endographite as a herbal drug to combine it with 5-fluorouracil as a marketed drug. So the nanoparticles are synthesized in order to achieve the synergistic anti-cancer activity. Endographite is isolated from Endographis paniculata. It is amorphous in nature and yellow color and odorless and extremely bitter. This is the mechanism of action of endographite. This is the overall schematic diagram showing research work performed. The nanoparticles of endographite and 5-fluorouracil were synthesized separately and characterized using zeta-sizer, TEM, DST, differential scanning calorimetry, and FTIR. Then the combination therapy was performed in vitro and in vivo. For in vivo, A549 cells were used and in vivo, bulb C mice was used. Lipids were selected based on pre-formulation study, and then nanoparticles were synthesized and characterized and in vitro and in vivo. For pre-formulation study, different lipids were used such as stearic acid, polyethylene glycol, palmitic acid, and cetyl alcohol. And it was found that cetyl alcohol was better and highly soluble in ethanol. So we chose this lipid. The nanoparticles were synthesized using solvent injection method. This is the size distribution of nanoparticles. Transmission electron microscopy. The image shows that the synthesized nanoparticles were below 200 nanometer in size. This is the DST thermogram of nanoparticles of endographite. The results showed that the solid lipid nanoparticles of endographite were encapsulated better, and 5-fluorouracil also. This figure shows FTI spectra of endographite, cetyl alcohol, endographite, cetyl alcohol physical mixture and formulation. FTI results also showed better entrapment of drug in the formulation. This is a stability study of nanoformulation that showed that nanoparticles up to 3 months the nanoparticles were normal in size. They show the yield. The in vitro study was performed in A509 cells. The results showed that the combination of endographlet and 5-fluorouracil showed better anti-cancer activity than the herbal drug. In vivo anti-tumor activity was performed in Balsi mice, and early acid cells were used to induce tumor in peritoneum of the mice. This is the dosing schedule. The results showed, the tumor regression parameters and lifespan analysis was performed. The results showed decrease in tumor size in combination treated mice, then herbal formulation, and lifespan analysis parameters showed that mean survival time increase in combination treatment, and percentage increase in lifespan was also increased in the combination treatment than the herbal drug. Additionally, the hematological parameters were also assessed that showed that the formulations were safe to use as there was minor variations in the red blood cells of the mice. Thank you. Biopsy mice. Common peritoneum. In peritoneum, early acid cells induced peritoneum tumor. Ma'am, only tumor regression parameters and lifespan were analyzed. Ma'am, only hematological parameters. Yes, ma'am. Any other queries? Solvent injection method. Okay.
Ma'am, my question was, what was the method used to produce nanoparticles? So she said that it's solvent injection method. I want to know that what's the process for this method?
First, we have to perform this lipid solubility study. Then we can find out that the lipid is better dissolvable in the specific solvent. After that, we have to dissolve this lipid in the solvent with the drug. And there are two phases: aqueous phase and organic phase. And in organic phase there is lipid and solvent, and in aqueous phase there is your surfactant and stabilizer with the water. Okay, so we have to solubilize the lipid with the your compound in the syringe, and then we have to inject this solution in the stirring aqueous solution on magnetic stirrer. Then after that, after you will inject it, then the nanoparticles will be synthesized after half an hour, 1 hour. Ma'am, lipid. Name, ma'am? No, ma'am, it is lipid nanoparticles. Yes, ma'am.
So I wanted to know that study about the effect in humans. Its effect in humans. Like, do you have any data
Data related to what?
in human cancer? And if so, then like as you said that chemotherapy it has many side effects. We all know that.
Yeah.
So is the herbal treatment that effective that it will overcome the benefits of chemotherapy? Like if a person or any organism is at a last stage.
At last stage, I can't say about at last stage because yeah, but we can try because traditionally, since tradition, matlab since ancient times, so we have used many traditional drugs also. So they have cured many diseases. They might be cancer also. So I think, in my personal opinion, I think um, it can also be helpful in that stage. But before that, we have to optimize the concentration. After that, we can say that human, this drug, this formulation, yes, it is applicable to them because the compound is herbal, and one of the compound is herbal and another compound is a marketed drug. We are using the marketed drug already in the market. For example, such as 5-fluorouracil paclitaxel. We can combine this drug, herbal drug, with marketed drug also. I have used only a single chemotherapeutic drug. No, no, no. Right now, I mean, uh, yes, ma'am. Thank you.
Thank you, Zoya. Now I would request to Dr. Saima Wajid ma'am to please felicitate Dr. Usha Kiran ma'am to chair this session. So this is end of our session. We are wrapping up, but we have to wait because in hall 2 there are sessions are going.
खत्म हो गया बेटा.
Thank you all. I would request to all of you for a coffee break outside the hall, and then please gather here again for valedictory function.
लैपटॉप की जरूरत नहीं है.
Assalamu alaikum and a very good evening. I'm sorry for keeping you all on wait, but as you know, it is a valedictory function, so thoda results announce and discuss karne mein it is taking some time. But anyways, let's begin with the valedictory function. Very good evening and warm welcome to all the delegates, guests and students who participated here for the DBT and DST SERB sponsored National Conference on Biotechnology for Sustainable Development and Human Welfare, organized by Department of Biotechnology, Jamia Hamdard. As I mentioned yesterday, that the Department of Biotechnology organizes such conferences to enlighten the young minds and promote the participation of students at all levels. But because of the pandemic and the critical situations, we are organizing this event after 3 years. And it was good to see the vast zeal and enthusiasm amongst the participants. Over the last 2 days, we have had deep insight into several talks related to sustainable development goals to help the mankind protect biodiversity and spur the economic growth. I would like to express my gratitude to the Honorable Director General of CCRUM, Ministry of Ayush, Government of India, Professor Asim Ali Khan sir, who shared his views related to sustainable development goals, and our Honorable Vice Chancellor sir, Professor M. Afshar Alam. And also, I'm thankful to IQAC Director and Dean of School of Chemical and Life Sciences, Professor S. Raisuddin sir, and none other than Professor M.Z. Abdeen sir, the Organizing Secretary and Head Department of Biotechnology, for gracing the inaugural session. Now, with great honor, I would like to welcome our chief guest for the valedictory function, Mrs. Geetika Joshi ma'am, to please come upon the dais. She is the Managing Director of Lecture Team Biotech Private Limited. Thank you, ma'am, for taking out your time for us from your busy schedule. We are delighted to have you here. Now, I would like to welcome the Director, IQAC, and Dean, School of Chemistry and Life Sciences, respected Professor S. Raisuddin, sir. Please come up on the stage. Next, I would welcome the Organizing Secretary and Head Department of Biotechnology, Professor M. Abdi, to please grace the dais. Thank you all. Now, I would request, oh, yes, I would request Dr. Reema Pant, ma'am, to please come up on the dais and honor us. Now I request the School of Chemical and Life Sciences Professor S. Raisuddin, sir, to please welcome our chief guest, Mrs. Geetika Joshi, ma'am. I request sir to please welcome our Dr. Reema Pant, ma'am. A token of appreciation for ma'am. Now I would request Professor M. Abdi, sir, to please welcome our Dean, School of Chemical and Life Sciences, Professor Raisuddin, sir. Now, in last 2 days, we had 11 plenary sessions from distinguished leaders of the field, like Professor Deepak Painter, Professor Sudhanshu Brati, and Professor S.P.S. Sanuja, 25 invited talks from eminent scientists, all sessions from different themes like sustainable agriculture, environmental sustainability, pharmaceutics and nutraceutics, intellectual property rights, entrepreneurship, infectious diseases, molecular medicines, and nanodiagnostics were very lively. All talks have focused on cutting-edge technologies and ongoing research in field of biotechnology to achieve United Nations Sustainable Development Goals. This conference showcased over 85 presentations from young researchers with 17 oral and 65 presentations which were nominated for award. Among them, the eminent judges selected 3 best oral and poster presentations along with some token of appreciation in the form of consolation prizes. Now, I request our chief guest and Reema Pant ma'am, our guest of honor, to please felicitate the winners. We have the winners for the oral presentations, and now I would announce the name of the winners. So the first winner for the oral presentation is Miss Asna from Jamia Millia Islamia. Sir, the second winner. The second winner in oral presentation is Ms. Qudsiya Ashrafi from Jamia Hamdard.
Congratulations.
Now time for the third winner in the oral presentation. It is Ms. Sonali U. Chandrashiva from SPPU University, Pune.
student of Professor Nadar? Okay. Now I would invite the budding researchers for the token appreciations. In oral presentation, Dr. Shoaib Alam from Jamia Millia Islamia. The next is Dr. Rohan Chabra from Prakriti Agritech Rohini. Dr. Rohan Chabra. The next winner is Narjis Sabah Khatun. Narjis Sabah from Jamia Millia. Okay, so we have some time for the winners for the poster presentation. So now I would request the chief guest and the chairpersons to please felicitate the organizing secretary, Professor MZ Abdeen, Head Department of Biotechnology, and the man who made this event functional. Now I would request the convener, Dr. Javed Ahmed Sheikh. I'm sorry, Dr. Javed Ahmed Sheikh, to please come Now I would request the co-convener, Dr. Javed Ahmed ma'am, to please come up on the Okay, she's finalizing the results, so I would request and invite the organizing committee for the felicitations. I would request Dr. Humaira Farooqi ma'am to please come up on the stage. Now I would request Dr. Saima Wazir ma'am to please come up on the Now I would request Dr. Nilaj Ahmed Ansari sir to please come up on
So we have the star of this evening. We have star of the students. It's Dr. Monica.
Now I would request Dean School of Chemical and Life Sciences, respected Professor Raisuddin sir, to please come up on the mic and share his views regarding the conference. Welcome, sir.
Very good evening. The energy level is very high. You have been offered very good food, tea, and other good protein and good vitamins and maybe fibers also. So, first of all, I must congratulate the organizing secretary, Professor Abedin, who is always he's one of the professors of the university who has very enthusiastic energy to develop networks with the industry and also with the research institutions. So because of his effort and his contribution in the field of biotechnology, he has been able to gather such a big gathering from across the country, from Jamia Hamdard, from Jamia Millia, Aligarh, Pune University, and so many other research and academic institutions. So, therefore, it has been possible, and I see very rarely that in the valedictory session, people are so much awake and they are still clapping with the heroes and heroines, which may not be taken in bad terms, but champions of the conference, like Dr. Javed Sheikh, Dr. Saima Wajid, Professor Dr. Humaira Faruqi, Dr. Miraj, and very enthusiastic and available and untiring energy and spirit, Dr. Monica Safi, and all other teams. And I attended very limited number of sessions, but I was having feedback, all the lectures by the speakers as well as by the delegates are very much, have been very much relevant to the field. And I wish that you go with a good message from the university that biotechnology is alive to the call by the society. All the major problems humans face, biotechnology have answer. It may be health, it may be disease, it may be environmental problem, environmental pollution. If you go to the environmental degradation, biodegradation, environmental remediation, there is a biotechnology. We have this problem of health and India in particular, lots of issues are there, especially the health conditions and stunting of the newborn is one of the issues. And I think recently Global Health Index has shown that our populations, huge number of population is having problem. And besides, I urge you that besides going to the academic field, very routine kind of career path you take, do MSc, do PhD, and become assistant professor and become further guide of the research scholars and finish your life. Why don't you take entrepreneurship in the biotechnology? Government has been offering incentives for for establishing small startups in any field you can start. And I have seen, I have met many people also. They have utilized this biotechnology knowledge to become the job giver instead of job seeker. This is the issue with biotechnology. Huge number of biotechnology seats are there in various universities, which produce a lot of biotechnologists. Sometimes without much, just for sake of their biotechnology. There are many, I know, Professor Abdin will vouch that a large number of students come for practical training in Jamia Hamdard. And on their prospectus, they write, we are number one in biotechnology. But at the same time, they don't have simple techniques. Sometimes I ask, do you have a spectrophotometer? They say, no, sir, what is a spectrophotometer? Because I am the controlling authority for training, administrative authority for facilitating training to the BSc as well as MSc students who come from other institutions So I sometimes interview them that what facilities, what have you seen pH meter? They have seen no, sir. I have been taught theory that there is a neutral pH 7 and how to use it. There are two types of pH meter, theory only. So practical knowledge of biotechnology, in that sense, our institution is very important milestone in the education of biotechnology. No other department has such number of seats available. For example, in chemistry, there are 25 seats, or biotechnology have 50 seats as against, and all 50 seats are sought after. There is a huge demand versus seat availability ratio. And the scholars as well as MSc, BSc students are performing very well, and they find very good environment in the Department of Biotechnology for research and training, and they shine on the origin of biotechnology. So this has been, so I must again congratulate the young energetic team of Professor Abdi, who has been working. You must be knowing that for some time Professor Abdin was not feeling well. For about 1 week he was indisposed, he was in hospital. So, still his team has performed and given and delivered the type of conference, the type of event Jamia that has been wanting. So I hope that your experience here for 2 days has been very good. I congratulate the oral presentation award winners and those who could not win. Don't worry, maybe the next opportunity will be there waiting for you. So somewhere, somehow you will again win prize. But do good research. I am also the Director of Research and Development Cell. The quality of research is very important for you, and good publications also. Always target high impact factor journal or very reputed journal. So as this one has helped us, Jamia Hamdard, as well as department. So there is a competition that who will publish paper in high impact factor journal. So from our department, many papers come with impact factor 11, 12, even one paper has come in 24 impact factor also from the university, and some good papers. All the faculty members have research projects, and one of the projects, very high value projects, is with Professor Abidin in collaboration with the industry, Hydro Green. Similarly, one project by India Alliance DBT Welcome Trust is with Professor Dr. Miraz Ansari Sahab, more than 3 times. He has spent just for 3-4 years only in the university, but he has made his mark, and he has project worth 3.5 crores from Welcome Trust. So such a big project. Similarly, in no other university we offer opportunities like that. As a being assistant professor, junior most position in the university, he has been given the responsibility to develop a department of medical microbiology, which he has done successfully. And many times we don't believe in that. If somebody is flourishing, even if he may not be professor, in many universities there is a system that पहले professor बनो, फिर head बनो, फिर in charge बनो, फिर ये बनो, वो बनो. So if somebody has a sparkle, he has potential, even being in a junior most designation of the university system, but we provide and we enable, we empower the research scholars as well as this. So these kind of good systems have been there, and Jamia Hamdard has one of the highest citation per faculty ratio, only next to the university. And those who are familiar, they know that in pharmacy is #1 in NIRF for 4 years, the last 4 years. So sometimes it is not like that one time treasure is struck. It's a consistent and sustainable performance of pharmacy faculty. And in that biotechnology is also. contributor in the sense that they have collaboration with pharmacy school, and there is a pharmaceutical biotechnology program with M-Pharma in pharmaceutical biotechnology, and many research programs are running between collaborative way between the biotechnology teachers, researchers, and the pharmacy teachers and researchers. Similarly, in biosciences also, we have performed very well. Professor Abidin has been listed amongst 53 biotechnologists or biologists in CC. Fortunately, I am also one of them, but I am not a biotechnologist, I am a toxicologist. Maybe they have recognized me also. So that way we go ahead, and I wish that Professor Abdin will make it an annual event. Why don't you make annual event? So in the next year again, you organize such a conference. You were discussing that he is planning to organize an international event, which he can, because I know he has international contacts and international relationships. And he has shown that biotechnology can go up to field also, especially his program with IPCA laboratories has been resulted in the IPR, also product development and project commercialization also. Artemisinin project, which they have developed nasal spray for artemisinin and has been marketed in, I think, Australia, I guess. So in the lab and in the field, this department is shining. I wish that they shine more and become role model for other departments. I invite all of you as a Dean Faculty of Science and Chemical and Life Sciences to visit laboratories and state of facilities you see. And if you want to collaborate with any one of the teachers, you can. And also I wish that those, if you want to develop, pursue your career, like Monica is postdoctoral fellow. So if you want to apply for postdoctoral fellowship of SERB, as a National Postdoctoral Fellow, or ICMR Fellow, Research Associate, or DBT Research Associate, and DST Inspired Faculty, Women Scientists, there are so many opportunities available for postdoctoral research. And UGC has also recently opened, but I think it's closed now. 10th October was the last date for Dr. Radha Krishnan postdoctoral fellowship. So those kind of fellowships, if you want to apply, this we will forward your application and recommend strongly, then you will have good chances of success after PhD. Okay. So with these words, I wish all the best to you. And I think oral presentation, poster presentation result is there.
I think we'll be announcing shortly.
ठीक है, तो बड़ी मेहनत करनी पड़ रही लगता है, कंपटीशन बड़ा टफ था.
Sir, there were so many candidates.
There were so many candidates and all must be good. So sometimes selection is very tough. So I hope they will be completing. So with these words, I wish you all the best. And I again must congratulate the team Abdin and his colleagues, which I mentioned already. Those who have left out, please pardon me. And with this, I say all the best to you. Okay, thank you.
Thank you so much, sir, for such kind words. Now we have the winners for the poster session with us. So I would request the chairpersons to please felicitate the winners. The first prize goes to Syed Masir Azim.
So he is a graduate student, undergraduate.
The next winner is Mr. Kaushik Haldar. Mr. Kaushik Haldar from Jamia Hamdard. third winner is Mr. Rahul Sharma. He's from IMM. Now, time for the token of appreciation. The first name is Rajshri Sanyal. Rajshri Sanyal. Left. The next one is Paramdeep Kumar and Mansi Dahalia. The left. The third one is Bushra Zabi and Firdaus Kamar. Thank you all. Now I would request the organizing secretary. Bushra, okay, she's here. Thank you all. Now I would request the organizing secretary and head department of biotechnology to please be on the mic, who without minding his health, try to make this event functional. So welcome you, sir, for the conclusion.
Good evening to all of you. I am seeing all the young minds here. It shows that our country is moving towards progress to the great future ahead. And hope that the budding biotechnologists will come up as an entrepreneur in future. Actually, I have took an adventure to go for a lecture in Dharamshala. It was an adventure because actually I was organizing this conference and definitely I thought that, okay, I will go and come back and then will, but unfortunately I could get ill and then admitted to hospital, but with the prayers, your prayers and prayers of our colleague and well-wishers, I am now here with you. I am really thankful to my students and colleagues and dean and other well-wishers who make this program a successful program. And really, I owe this to all the sponsors who give us generous donation, including government agencies. I never thought that we will be able to get this much fund, but anyway, with the grace of Almighty, we got it. And we have been able to organize it. Actually, we thought this conference when I take in charge as head of the department. During my tenure as head of the department earlier, I was organizing the conference annually. It was an annual event. We have also organized workshops and many other things. So because of the COVID and then I was also not getting in between. But anyway, I was definitely in the department and I also wanted that we should organize. But because of COVID, actually, we could not be able to do it and it was discontinued. But definitely, as our dean has said, we will going to organize it again on yearly basis. But anyway, I have already discussed with our Honorable Vice Chancellor and other authorities of the university and then various sympathizers and collaborators who are with me. we will be able, inshallah, to organize an international conference in 2024, maybe early 24 or in the late 23rd. So that is my promise for the department and for the school and for the university. We will be able to organize it, and we will establish our secretariat very soon for that. Actually, I will not, I cannot make whatever we did because of the cooperation, because of the help and support that we got from our faculty, Dr. Javed, Dr. Saima, Dr. Humaira, and Dr. Miraj. Actually, once I thought that I should postpone this conference, but they said, "नहीं, sir, हम लोग जो है मिल करके करेंगे और जो है कोई दिक्कत नहीं है." वरना जो है when I was in hospital, I told my daughter that you should contact Dean and our colleagues that we should postpone it at least for 15 days. But they said, "No, we will do it," and they did it. They live up what to our students, especially I thank Dr. Monica, Kusia, then Sadia, Firdaus, and especially I thank Usha, and then all other research scholars of the department who really worked tirelessly for the success of this conference. Hope that you have already enjoyed it, and those who are coming from outside and also from different parts of the country, from Delhi, if there is some problem or some difficulty you have faced, I'm sorry for that. I apologize if there is anything, and hope that in future we will further do work and do improvements and will facilitate you so that we live up to the expectations of our great founder, Janab Hakim Abdul Hamid Sahab. Actually, he was engineering, and he established this institution from his own money, whatever he earned. He envisioned that how we have to make the people who are living in this country, how we can empower, how we can serve them, make their health better. So in that direction, I think we have to also work and help people to come up. And actually, the title of this conference, I would like to say that we together sat and then discussed and decided that sustainable development is the UN Charter, and that has been made in 2015 and then accepted by the leaders of the different countries. So we thought that why not this should be biotechnology for sustainable development and human welfare. So we did it. Thank you very much for your help and support, and hopefully in future we are going to meet again here for another good conference, discussion, deliberations, and then coming up with a solution to the humanity, not only for the people of country, but also the humanity, which is facing grave threat from what we did in past. Actually, as Dr. Vimapan yesterday was telling, the nature rejuvenates itself. When you burden nature, then nature start working how to rejuvenate. And we are now getting signs of those in terms of tsunami, in terms of flash floods, then many other things, volcano eruptions, typhoons, then many other problems, including what we did in the past. So we have to now wake up and do ourselves. We should rejuvenate the nature, the earth, to save this planet. Thank you very much.
Thank you so much, sir. Now, I invite feedback from the delegates, guests, and students. We would love if you would share your views about the conference, your stay here, about the lectures, how you felt, or anything, whatever you would like to share with us. Please come up with something. Anything, anything. Please. Any student, any participant, any experience, just feel free.
Good evening. I have a very small feedback to give. The feedback is that it is one of those very amazing conferences where I found all the students so disciplined. Usually colleges are places where people are in their own world, but amazing. All of you were so well behaved. It actually seemed like a school to me. And I was often wondering whether I am in a school where everybody is so silently walking in, sitting, listening very, very patiently to all. And I know post-lunch and post-tea sessions are usually heavy, but your spirit was amazing. So that is wonderful. I think people who are so patient are actually good scientists and researchers because science needs a lot of patience, a lot of introspection, a lot of quick thinking, and sometimes a lot of alternative thinking. Sometimes we are sitting patiently in a pond which has no fish, so we will never get a fish. And maybe we have to change the perspective, change the direction, we might get a pond with a lot of fishes. But again, for that, you need little patience and insight. So wonderful people, all of you. So, sir, I would really thank you for inviting me here and giving me this opportunity to interact with all of you. Thank you.
So really a very nice experience attending this offline conference after the COVID. And as mentioned by Hanson, that sustainable development, yes, this is the requirement. That sustainable development in terms of environment, in terms of energy, in all fields, especially this biotechnology is helping lot for these upcoming diseases and all. So, and the energy I experienced here while reading this Dr. Javed, Dr. Jagri, Dr. Simon, so wonderful team. Without such a wonderful team, it's very difficult. So we are really confident. So thank you, team sir, all the respected guests of honor. Thank you very much.
Thank you so much, please, just your views, nothing much. Just your views.
Entrepreneurship आपने जो है कैसे शुरू की, you are an entrepreneur.
So it's a long story.
थोड़ा सा जो है brief में जो है बता दीजिए 2-3 मिनट.
Good evening, friends. Yeah, it is true that me and Dr. Rima, we were talking about the discipline and the quietness, the seriousness among all your students. So it was really great. And we're talking about that, the same thing that she has described. So, well, I am, I have a very different background I come from. That is what Sir is saying, that I tell that how I started my business, to tell about you that. So, see, we say that there is called destiny. I wanted to become a doctor. And I was a good student, I would say. I studied with my scholarship and all, I got scholarships. and topper in my district and all those things were there. So I was very confident that I will definitely clear my, that time we used to have CPMT, the UP board, the Uttarakhand was not separated and I belonged to Uttarakhand. But unfortunately I worked hard and obviously I was very confident that I will clear. But destiny is something else for me. In fact, after immediately the exams, I packed my books, gave it to other students because I was so confident, that I have anyway going to clear it. So the time when we were supposed to get our result, the news came that the results were leaked, the papers were leaked, so the results will not be valid. So there will be re-examinations after 3 months. So that was the first shock that I came in my life, that okay, this kind of thing also happens. So कहते हैं ना दुनिया से विश्वास ही उड़ गया था, ये ऐसा भी कुछ है, मतलब पूरा विश्वास उड़ गया था. Because I studied science and after that I told my father, I said, look, come what may, I am not going to study this science again, it's enough for me. And he said, करना क्या? He said, then you do, तुम पढ़ने में ठीक हो, so you do your BSc, MSc, PhD and become a professor, get married to, because my father comes from the army background, get married to an army guy, it's a safe thing, मैंने कहा इतना नहीं पढ़ना है. So then he said, when you do a B.Ed, become a teacher, that's again is a safe job for you. But trust me, there was something going in my mind and I told my father, I said, I want to do something new in my life. He said, what new? I said, I want to do BA. That point of time, when we were, I think it was 1992 or 93. Anyone who is very intelligent, studious, it's a science. वो तो चिपक करके आता था कि भैया science ही पढ़नी है. If you are studying arts, so you are like a very stupid kind of a person. So my father did not agree. And he said, no, nothing doing, you have to study science. And trust me, every day I used to pray that someone should take me away from this house or I will run away. But वो possible नहीं था run away करना because obviously there are many issues. So fortunately, we had some friends from Delhi and his wife was a fashion designer. The word fashion designer was very fascinating, at that point of fashion designer and something new, something interesting. So she told me, what you are doing? I said, nothing. He says, why don't you come to Delhi? मैंने मनी में सोचा कि oh, this is what I was thinking. I said, can I come with you? Will you take me? He said, हाँ चलो. मैंने कहा मैं करूँगी क्या? He said, we will see, we'll figure it out. You can also do fashion designing. I said, are you sure? He said, हाँ. I came to Delhi and trust me guys, I have 75 rupees only with me. And I have one suitcase with half clothes here and there. Something like, kurta hai to pajama nahi hai, something like that. And I came, but my parents, this whole plan we made in the night and next day morning, I was with my suitcase. My parents said, where are you going? I said, I'm also going with them. So I came to Delhi only with 75 rupees. And from there, the struggle started. My first job started with 600 rupees. My parents thought कि कब तक survive करेगी, पैसे तो नहीं है तो वापस तो आएगी. But I was very clear I'm not going to go back. It's 2 years my parents did not talk to me because they did not agree that what you are doing over here. So after that I started working better. Then I did my own studies. I did my fashion designing. It was a lot of struggle, but I was very clear with one thing, that whatever the industry I will go, I want to reach at the top one day. 2002, I started my business, but the passion was there that, I want to do something on my own. I learned the industry very well, worked very hard, day and night. Someone was saying in the conference today that you work 15 hours. I said, I still work 16, 17 hours, so that's what at the end of the day it pays actually. There is no rocket science to success. There's no rocket science. So I built up my business and trust me, that point of time when I got this opportunity to start my business, I had come across with a Spanish company and they wanted me to represent them in India. So I told them, guys, I don't have money. This is my background and I don't have money. I said, what I have is my hard work and honesty. That's the two things I have. Money I can earn. They invited me to Spain First time, sir, I don't have money to go to Spain. But जिद तो थी कि जाना तो है, opportunity खोनी नहीं है because सुना था, पढ़ा था कि opportunity comes, doesn't knock every time. So trust me, I only had money for the ticket, and I only had 12,000 rupees to go to Spain for a week. And I did not tell anyone that I have this little of money with me. I went. ऐसा देखा जाएगा वहाँ पे जाकर के क्या होगा, खाना नहीं खाएंगे, देखा जाएगा, रहने का भी देखा जाएगा, कुछ नहीं किया था, ना होटल था, ना कुछ था, ना कुछ था. And Spain, that point of time, was a country where people hardly understand English and all. I went, and trust me, guys, I think 3 nights or 4 nights I slept in the railway station. I used to eat only one burger the whole day. That was only and only my meal. Used to have the public toilets. So that was a struggle, But then I built up the business, and I built up a business from a garage, and I built up a company of 150 crores with a 3-star export house. And that we get the star, like when we do the exports, we get the star rating, and you have to maintain your turnover every year by year. So definitely after COVID, we changed our line a bit. We toned down the business. Now we are building it up again. And the export also, we are going towards the sustainable fabrics and all. As the hemp fabric, as you have seen outside, we are working a lot on that. And we are also, now we don't want to go on the commercial, these big machines and things, big factories and all. We are not going into that. But we are working on trying to work more on the villages, of the Uttarakhand, more interior villages, and trying to build up the base over there so that we can give the livelihood to the people over there as well. So what I can say that, to the young generation, that because I have a lot of, every year we have young people coming in our offices. And what I see that, every year by year, the pattern is changing, and the pattern is not changing for the good, because people they want to work less, but they want more. It's not possible. If you go down the line 10 years later also, the balance will always be there. You work hard and then only you'll get it. So there are a lot of shortcuts happening, which is not good. So if you have to make your base strong, your career base strong, so the base strong you can only make by the hard work. And there is no shortcuts in life. There's no shortcuts. So today, if I'm standing here, today we work, and I would say that, there's a long story. Definitely, I always love to take challenges. So there was a point when once I went to the Zara office for the business, they literally put things on my face. They says, "Are you stupid? You have come with these things. We don't buy these kind of things. You don't know what we buy. We are a fashion-forward company and this and that." It's like, what I did wrong? I think everything was fine. But, by the time, I could not realize that how forward they are. And then I started thinking, I said, okay, in 2 years, I want to be the #1 supplier out of India. And I became the Zara Kids #1 supplier. So we are the only supplier till 2017 who was producing the baby wear out of India. Otherwise, none other factory was able to achieve their quality parameters. But I was very clear that when someone from Portugal or from other countries can do it, then why we can't do it? So it's like that fire that, I never want to take a failure. So I always keep fighting. I always keep, looking for that, working on that no, I will never fail. I will never fail. So now we are working on the hemp project with Sir. So I told Sir that, because right now I'm busy with something else. I said, probably from December 1st week or 2nd week, I will come here every once in a week to understand and to learn more things from you. So Sir is going to, and Monica is going to teach me. So this is what I wanted to tell you all. So just work hard, and there's no shortcuts in life. Thank you.
Thank you so much, ma'am, for such motivational and inspirational words. Now, last but not the least, I would like to call upon the stage Dr. Javed Ahmed Sheikh, the convener of this event, for the vote of thanks.
So, honorable guest of honor and chief guest, the dignitaries on the dais, and our most favored the guests and ladies and gentlemen. It's my utmost privilege and honor that I have been asked to propose a vote of thanks for joining us for these for this scientific conglomeration. So I, on behalf of the organizing committee, Department of Biotechnology, and the entire fraternity of Jamia Hamdard, here together, and on my own behalf, a very hearty vote of thanks to all the speakers for gracing us with your important work and sharing us with your findings and the opinions today and yesterday. So to begin with, I would like to thank our esteemed Honorable Chancellor, sir, under whose umbrella we are tenaciously working towards the betterment of the human welfare. His undeserved trust and unequal support always allows us to perform by leaps and bounds in the field of academia, research, and medicine. I would also like to extend my thanks to Honorable Vice Chancellor, sir, Professor Afshar Alam, sir, under whose leadership and administrative prowess Jamia Hamdard is excelling and is at very good position today and will excel by leaps and bounds, inshallah. And of course, a very big thanks to our revered Professor Raisuddin Saab, the Dean SCLS and Director Research and Developmental Cell, and whose guidance and support, without whose guidance and support, such a grandeur event would not have been possible and ultimately reached a fruitful end. So we will always be indebted and look always for your patronage in future. Further, I would like to thank our beloved Professor M.Z. Abdeen, the head department of organizing this program and organizing secretary of the event, the man who made this event functional with this constant help and guidance and radiated the source of energy with us without minding his own health. So we will always remain grateful to you, sir, and thank you for being with us always, and we pray for your good health. So a word of special thanks to my colleagues, Dr. Humaira Farooqui, for the help that we received towards this conference from time to time, guiding us with whatever and whenever it was required. And I'm very grateful to Dr. Saima for her constant support and for looking after the cultural event. And sorry for disturbing you at every moment. And sincere thanks to Dr. Miraj Ahmed Ansari for his help and support at various stages for coordinating and managing the hospitality and management team so well. I'm grateful to Dr. Jagrati Narang for being the co-convener of the event and for always being there and helping us in making the event functional and managing the food committee so well. And importantly, this event would have been charmless without the management and the anchoring prowess of Dr. Monica. So a big round of applause, please, for Dr. Monica. So I'm very much thankful to our current and former faculty colleagues and non-teaching staff members who have always stood by us and motivate us. And I feel proud and thank you for making this event a successful one. A special mention I would like to make up Mr. Anish for all the secretarial assistance that he has provided, and the other persons from the staff who have always helped us, like Mr. Islam, Mr. Imran, Mr. Wajid, Murad Saab, Satish, Nazir Saab, and Shafuddin Saab and Mubarak Saab. So an event of this dimension cannot happen overnight. All of you know the wheels start rolling months in advance, and we have been fortunate to be backed by a team of very motivated and dedicated researchers and students. You will see all of them here. I would like to mention the volunteers. I would like them to stand up and have a round of applause for them. Kutsia, Firdaus, Anuradha, Priya, please. Sadia, Anupam, Anand, Jasmine, Nigar, Sharik, Rebecca, Rahim, Wasim, Furqan, Anam, Aditya, Manu, Afreen, Sanskrit, and Rupali. They have tirelessly worked for this conference. Thank you all. Well, honorable guests, an event like this cannot happen like on such a pace. So the process takes weeks to plan and requires attention to the tiniest of the details. For that, we have been fortunate enough to be backed by a team of very motivated and dedicated volunteers who performed the assigned duties so diligently. I cannot thank everyone for their involvement, for their willingness. Please forgive me for that because there have been many volunteers. I would like to have the name of the teams that we had, like the team for the travel and accommodation, and Dr. Akram was looking after that. Can we have a round of applause, please, for the team? It's for the whole of the team. I am thanking you. The team at the registration desk, please, thank you all. The team with the dice management that managed the halls beautifully, and the food committee. And most importantly, to a big thank you to all the students who were with us for these 2 days, tirelessly here. I might have missed many of the acknowledgements, so please forgive us for that. And finally, I would like to thank the national sponsoring agencies who have sponsored this event, the DBT and the DST SERB. It would have been impossible to organize it without their support. And a big thank you to the industrial partners like Sheen Biotech, the Biorad, Imperial Life Sciences for providing the financial stay. And I would like to conclude with the word that if time would have been money, you have literally spent millions with us for these 2 days. So I hope you have enjoyed the event. This event would not have been successful without you. On behalf of the organizing committee, I'm very grateful to all of you. So for being with us, God bless you. Thank you.
पंजाब सिंध गुजरात